virtual, please be aware of your time, otherwise we're going to have to interrupt you. So that's all the minutiae. I do want to add my welcome to everybody in the room and thank you for your participation and attention. And I know that the students are super excited to share their work. So I think we're actually right on time. And first up, we have Jessica. Jessica is an intern with the Pacific Islands Fisheries Science Center. She's a Pacific Young Scientist Opportunity Intern with the Pelagic Research Program. The title of her talk is It's All About Plankton. And you have the mic. And exploring the exploration of the zooplankton communities in the North Pacific Ocean.
Aloha kako, my name is Jessica Lee Aquino, and I'm a former Leeward Community College student and a University of Hawaii Manoa student. So being born and raised on the Leeward side of Oahu, I've always been fascinated with the outdoors and the world around me. So with this PYSO internship, I was able to delve deeper into my curiosity. That being said, I'm going to talk about the exploration of zooplankton communities in the North Pacific Ocean. For my internship, I got the opportunity to work with plankton samples collected from the Big Eye Oceanography cruise last year in 2022. This cruise sampled the physical, chemical, and biological habitat characteristics of our waters. So the survey also collected scomber larvae, which could also help researchers figure out where Big Eye and other tuna spawn. Likewise, my objective is to establish an environmental gradient and species distribution model for Big Eye tuna. Why zooplankton? That being said, zooplankton is an essential ecosystem functioning because they provide food for higher trophic levels. Because zooplankton biomass estimates available food, this may predict areas with more larval fish recruitment. Likewise, zooplankton populations are also early life stages of species that are commercially important and conservation importance. The research focuses on describing population and stock structures, as well as better understanding the variability in the North Pacific ecosystem that influences recruitment and distribution of Big Eye tuna and other highly migratory species. In-field data collection. This transect crosses the North Pacific Gyre and it spans a steep gradient in subsurface ranging from relatively warm and oxygen-rich waters of the subtropical gyre to cooler oxygen-depleted waters closer to the equator. So this transect corresponds with a gradient in catch per unit effort in the deep-set longline fishery, with high Big Eye tuna catch rates in the north and relatively low catch rates in the south. With these broad biogeochemical and fishery gradients are reasonably well described, that link between oceanographic properties and fishery productions remains limited. So this survey aims to collect data on lower intermediate trophic levels to help bridge the knowledge gap between the ocean, biogeochemistry, and the region's top predators. So as you can see, the stations plotted on this map on the right-hand side is highlighted Station 6, which I worked solely on. So it's right in the middle of the transect lines. Thank you. And the CTD rosette collects conductivity, temperature, and depth, and the 2-meter ring net was the plankton samples, how they were collected, and the ones I was working with. So lab data collection. Sorting fish larvae out of zooplankton is a challenging task due to their small size and similar appearance of these organisms. In my case, I utilized the dissecting microscope to sort the zooplankton visually. But this is time consuming and it requires a lot of expertise in larval fish identification, but it's effective in locating specific target species such as Big Eye tuna. So as you can see in the circular picture, this is the type of sample I was working with, very thick, but I sorted out this whole sample in about 4 weeks for that whole jar. So about 40 working hours, so each week, just looking at the sample and sorting it in those well plates. So getting into identifying fish larvae involves examining their size, shape, external features, and in some cases, internal features. Key characteristics such as fin rays, pigmentation patterns, mouth shape, fin position are important for species identification. With that being said, I use taxonomic keys, field guides, and expert help from my scientific advisor, Bruce Mundy, to help me with the challenging task of identifying these fish because of their changing morphology and rapid growth rate. So I chose these pictures to illustrate what can be in the sample. So it's a variety of things, even though Katsuana polemus, the first picture, is a type of scomber larvae, which is tuna. So that's kind of the target, but I didn't come across of any big-eye tuna yet. But as you can see, we have some cephalopods, fish eggs, and that's just a little bit of what you can see on the top is the well plates of all the species I identified. So the density sorter. So as a result of the thick plankton samples, I decided to switch gears and focus on a density gradient contraption with the help of the Maker Lab team from SOD and my mentor, Dawn. I was able to construct a device to help sort the zooplankton. This technique separates the zooplankton based on their different densities. The density gradient solution I used was a high sodium solution, and it helps separate these fish larvae or zooplankton into two distinct layers. So gastropods like this will sink down to the bottom, making it easier to sort out the heavier, more dense items. And in the big picture to the right, I have the density sorter that I created with the help of the Maker Lab and Dawn. So I'm also using environmental data summary for my project. What is EDS, you might ask? EDS is an R-based program that can handle gridded environmental data at different temporal scales. EDS can also download and extract temporally summarized satellite data, oceanographic data by any point in time with the use of advanced very high resolution radiometer and VIRS, visible infrared imaging radiometer suite. So these collections where they just take high resolution photos and you can basically get data from space. So you can get sea surface temperature, wind speed, chlorophyll A just from space. And you can access this information using EDS when you're coding with R. So you might be wondering how does fish larvae relate to EDS? Fish larvae are highly sensitive to changes in environmental conditions, and EDS tracks this environmental variability, including fluctuations in temperature, currents, nutrient levels, and these all can impact larval survival rate and growth. So these three map plots that you see are good data contrast. As you can see on the legend on the right-hand side, there is a color gradient and the changes in spatial patterns as you move north to south. So when looking at SST or sea surface temperatures, you can see that it's much warmer in the south and cooler in the north. And going to chlorophyll A in the middle, you can see that chlorophyll is higher in the south, as well as wind speeds higher in the south as well. So for my next steps, plans moving forward with my PYSO internship. So what I plan to do is finish sorting all of the plankton samples so I can develop a species distribution model, as well as getting a better idea of how these environmental changes and variabilities can affect fish larvae such as the big-eye tuna. And what am I going to do with this? So this approach aims to understand the interactions and incorporate them into management strategies by addressing the importance of big-eye tuna in fishery management, stock assessment, ecosystem-based management, and research conservation. Thank you. Any questions?
So I have a question for you. What surprised you? So I did a lot. I did hundreds of hours of this kind of stuff for graduate school. So what came as a surprise to you?
What came as a surprise for me is having all these resources and scientists around me so I can ask them questions whenever I needed to, and even being able to utilize EDS and R and learning how to code, as well as using the information available to me, because I didn't know that you could get data from space and just code with it in R. So that really surprised me. Thank you.
Thank you. So I would like to invite Joy Smith up. She was your mentor, and she wants to say a few words.
Great job, Jess. Thank you. We're so excited to have everybody here. First one's done. What a good job. We're so proud of you. As Jess said, she is transitioning now from Leeward Community College, and she is about to start her program as a bachelor student at California State University at Monterey Bay to study environmental science, technology, and policy. She came into this project having experience from the community college, but also from working at UH Manoa, having worked on samples with plankton from both Pearl Harbor and Kaneohe Bay. So when she joined this project, she could jump in and really take her experience and ideas and really participate in a influential way. And we're really thankful for her mentor team, which was myself, Kisei Tanaka, Johanna Ren, and Don Kobayashi, but also John Whitney and all of his lab mates, who this project was part of a bigger project, and they all really helped her as well. So thank you and good job.
Thank you so much. Thank you, Jessica. Really appreciate those remarks. So next up, we have Nick Beltramini. Am I saying that correctly? Nick is a hauling scholar with the PIFSC Bycatch program, and he is mentored by a not swimmer. The title of his talk is Sharks, Skates, and Rays, Bycatch Mortality, a Literature Review.
Thank you.
Do you know what is that?
Yeah.
Okay.
All right. Well, first of all, I want to say I have so much respect for Charles because he actually admitted to using ChatGPT. I wasn't going to tell you guys that I did too. But may have or may not. I grew up having this impression of sharks that there's a sense of invincibility with them. They're the top apex predators. They roamed our oceans with grace and power, and were fear and awe-inducing. Yet when I started this internship, I quickly realized that our shark populations are extremely vulnerable and fragile to overfishing pressures. Sharks are caught as bycatch, which means they're not targeted and they're actually caught accidentally or incidentally. So if I go to catch a tuna and I reel on a shark, that shark will be bycatch. These are 5 fisheries in the US that have shark as bycatch. They're not all encompassing. These are just 5 primary fisheries that I found throughout this project that tend to have, high rates of bycatch mortality. Pelagic longline, bottom longline, purse seine, and gillnet being commercial, and rod and reel being a recreational fishing sector. For the purposes of time in this presentation, I'm only going to be talking about the pelagic longline and bottom longline fisheries. And what I did was using keyword searches, using friends and mentors and colleagues at NOAA, I was able to find as much public information on the internet and also unpublished data and just raw observer data to pool together all different gear types and all different species of both bycatch mortality at vessel and post-release. So there's 2 types of mortality rates that the literature talked about, one being at vessel. So an at vessel mortality instance occurs when a shark is hooked. This is a bottom long line. And by the time it gets up to the vessel, it's already dead. And then it's discarded and goes back to the bottom. That's an example of an at vessel mortality. Post-release mortality, on the other hand, is a little bit more nuanced. And this is when the shark actually comes up to the vessel and is still alive. And the only way to monitor exactly and directly how it's going to fare after it gets released is to tag it. And these tags usually range over $1,000 each. They're satellite tags. So imagine taking 1, 2 of your laptops, equipping it on the shark, and then throwing it overboard. And then we can go ahead and actually monitor how they fare post-release. And a post-release mortality incident usually results in the fish dying and sinking to the bottom. Here's an example of a satellite tag. If you take at vessel mortality, combine it with post-release mortality, then you can get the number that fisheries managers want to know, and that's the total amount of individuals that die when they come in contact with fishing gear. And that's the number that I'm trying to find today. In terms of that vessel mortality, I saw 49 papers that encompassed over 1 million individual fish. So this is coming from a lot of raw observer data, which you can have such a high sample size. And it encompassed 90 species. And this is a representation of that finding. Each dot on this graph is its own separate study, and the size of the dot is relative to the sample size. So a larger dot, like you see here on the blue sharks, encompassed over 30,000 blue sharks and said, as a result of these 30,000 blue sharks that we have access to, we say the at-vessel mortality rate for pelagic longlines is roughly 15%. And you can see that not only between the species, but also within the species, there's a huge range between what these studies are saying the at-vessel mortality rates are. This is a bottom longline fishery. And notably, you can see dusky sharks literally range from a mortality rate of essentially 100% die by the time they get to the boat to literally 0 or negligible amounts of fish die when they get to the boat. So that's important as managers to find out what's making these differences and to go into the methods of the study. Water temperature, size of the individual, sex, circle hook versus J hook, the size of the hook can all have a huge difference into what these at-vessel mortality rates are. And it's important as fisheries managers to know how they can influence at-vessel mortality rates. And it's also very important as managers not to rely on single data points and single studies to find what these model at-vessel mortality rates are. For example, if we take this study that says the at-vessel mortality rate for sand tiger sharks in the bottom long line is essentially 0, we could be grossly underestimating what the actual at-vessel mortality rate is just because of a lack of knowledge and research. And that's where the importance of a meta-analysis and a meta-paper comes in, where I can try to pull together all different resources to try to find the closest estimate. These red triangles are a weighted summary. So the triangle basically summarizes each study. If there's a larger sample size, it's going to have more weight into where that triangle lands. Now, it's even more important to go to the meta level when dealing with post-release mortality rate, which is, if you can remember, when you catch the shark, you have to equip them with a $1,000 satellite tag. And the only way to have a valid enough sample size is to pool resources in from a bunch of different, even countries. Can you release it? And then you can monitor its behavior. Usually they sink to the bottom, but this one, I can't even tell you how long that took me to do, but it worked. Okay. Roughly about the same amount of papers, but instead of 1 million individuals, now I'm only working with about 2,000 individuals. And that right there is over $2 million worth of tagging equipment, just to put things in perspective, and across 24 species. You can see here that there's a lot less data to begin with, but also it's still fluctuating. Here's the bottom long line graph. Again, within the different species, there's quite the range between post-release mortality rates. Some individuals can fare very well dealing with the stresses of at-vessel and post-release, some cannot. Another observation is that post-release mortality tends to be a little less severe than bottom longline mortality. And this is pure speculation, but in the bottom longline fisheries, typically the leader between the actual line and the hook is a little bit shorter. And if you think of sharks being ram ventilators, they have to move to obtain oxygen. And if they don't have the long enough leader like they do on pelagic longlines, they're not able to swim and get the oxygen. So I think that could be why we see bottom longline mortality rates being a little bit higher. Anyways, to put the two together, to combine at-vessel mortality rate and post-release mortality rates, this graph shows the percent estimates for each species that they're going to die if they come into contact with fishing gear. So for instance, if I'm a silky shark, there's about a 55% chance if I bite a pelagic longline hook that I'm going to die before I even get to the boat, and about a 10% chance that I'm going to die if I make it to the boat and then get released. The numbers you see on the bars are relative to the amount of sample size. So there was 11,001 sandbar sharks that went into this number and 150 sandbar sharks that went into this post-release number. The range you see, like short fin makos, this is not very statistically significant because there's only 16 total samples. But this is just a testament of these species are not caught as often as, say, sandbar sharks or dusky sharks or blue sharks. So the last graph I want to show you guys is the proportional contributions to mortality rates. So for blue sharks, we can see roughly a little bit more than half of them die before they even get to the vessel, and roughly another half die after they get caught and released. And this is important for managers to know where we should allocate our resources to. We could theoretically be saving, 1/3 of the school sharks that are listed as vulnerable, or the oceanic white-tip sharks, which are on the Endangered Species Act. We could be saving roughly a quarter of them if we allocate our resources to safe handling practices and techniques by the fishermen. And for, say, silky sharks, which 90% of them are toast by the time they get to the boat, what can we do to enforce circle hook mandates and change the gear types that these fishermen are using to capture these fish? So with that, with my estimations and recommendations, I hope that we can make the ocean sustainable for all. And I hope to preserve the mindset and impression that I had of sharks, that they are the invincible creatures that I thought they once were. So thank you to your Not Swimmer, Toby Curtis, Heidi Dewar, Brett Cooper, Keith Bigelow for being my on-site sponsor. I would not be here if not for you. And all the fellow Hollings scholars for pushing me and inspiring me to do the best work I can.
Any questions for Nick? Yeah. What were your spatial parameters for this research? Like, was it only the ocean, or is it global?
It's global. Just the lack of resources and the amount of sample sizes, I have to literally pull from, Spanish studies, Hawaiian studies, Atlantic studies, just to be able to get an estimate like this. Yep.
These meta-analyses are so crucial to synthesizing what we know about this topic. Any other questions? Oh, go ahead.
Wait till the mic if you can have questions. So that people might start.
Great presentation, Nick. Do you happen to have timestamps on those studies? Just because for some of the work we do at PIRO, we're interested if there's a new, regulation on the gear, we want to see how the mortality changes between previously and then after that regulation.
Yeah, excellent point. I did log the time, and fascinatingly, the mortality rates tend to generally go down as we go on time. Pre-2000s, we're seeing mortality rates a lot higher, and a lot of these studies with these high mortality rates could be dating back to the early 2000s, when the fishing gear was drastically different than what it is today. So that's a good point. The continuation of my work, I'll definitely make maybe some regression lines of over time analyses.
Any more questions? Great. Thank you so much, Nick.
Thank you.
So next we have Derek.
Sprincis?
Derek Sprincis, and
Yep,
he's going to be
sounds
speaking
good.
to us virtually today. He's a hauling scholar with NOAA's Ocean Service, Hawaiian Island Humpback Whale National Marine Sanctuary, and his mentor is Patty Miller. The title of his presentation is Increasing the Environmental Resilience of the Hawaiian Islands Humpback Whale National Marine Sanctuary in Maui.
Sounds good. And isn't my expectation I should share my presentation at this time?
Yes.
Great. All right, are you all able to see my screen at this time?
We are.
Great. Well, good morning, everyone. I'm Derek Sprincis. I'm an engineering student at the University of Virginia. But before we get started, how many people are in the room, would you say?
Maybe 30?
Okay, that's pretty good, pretty good. So if I mess up, just think I'm an engineering student at Virginia Tech, not UVA. But we'll go ahead and get started. So today I'll be talking to you all about the objective of my project, give you a little bit of background, as well as actually give the results and a summary. So the objective of my project is to increase the environmental resiliency of the Hawaiian Islands Humpback Whale National Marine Sanctuary. And it was suggested by site leadership that the site is vulnerable to coastal erosion and shoreline creep, flooding, and sand encroachment. So the sanctuary itself takes up waters around all 4 of the major Hawaiian counties here in the state, with the major waters surrounding Maui County and the headquarters facility, which is what I looked at, being in Kihei, Maui. As you can see, the little blue dot here, that's the location of the town where the sanctuary is located. And here's a overlay Google Maps snip of the sanctuary facility highlighted in blue. Note that it's right next to the coast and it's surrounded by county land, which is going to become relevant later on. So the first problem that was identified, or possible problem, was erosion control. Now, I actually found this to not be a major issue, mostly because of a seawall that's been in place for the past 10 years. So you can see the images on the right side of the screen here. The bottom one, this one, taken in April of 2013, and this one taken just last month. There's been very minimal shoreline erosion over the past 10 years since this seawall has been in place. So I've just determined that this is not a major issue. That being said, some additional solutions that can be added or implemented to prevent further erosion, maybe over the long run, so like 50 years plus, is beach nourishment. Granted, this might not be the most practical choice because of regulations surrounding the area adjacent to the sanctuary. So surrounding the sanctuary, we have this very cool cultural site called a fish pond. Fish ponds are are traditionally used as a native Hawaiian aquaculture technique where fish would swim in and they'd be trapped, and native Hawaiians would be able to harvest them. Today, this particular fish pond is not in use, but it is still a cultural artifact and one that's being maintained and under very special regulation. Specifically, the regulations stipulate that modern equipment cannot be used within the vicinity of the fish pond walls. So that means that mechanical dredging can't take place, and that's typically how you would accomplish beach nourishment. Furthermore, the fish pond is actually a blessing overall, mainly because it serves as a breakwater, breaking down and reducing the erosive wave energy that may come in towards the shoreline. Furthermore, the fish pond is not a federal responsibility, and it's maintained by a NGO, so it's not necessarily something that NOAA can target directly, that is. So another problem is sand. So sand is blowing from the north onto the sanctuary property, especially the parking lot, overall just making the sanctuary look rather dilapidated and run down. Being here for the past few weeks, I can say firsthand that the sanctuary staff is very dedicated to making the sanctuary look good. But just because of this high winds and the sand and the beach close to the sanctuary, this problem becomes a daily occurrence and something that cannot be prevented easily. It needs an engineering solution. So I have 4 solutions that I came up with. One, either all 4 or just one or a combination of more than one can be implemented to solve this issue. For one solution idea is adding shrubbery along the sand dune approximately in this area. This would increase the potential for the sand just to stay in one spot. It would decrease the the ability for the sand to sediment move from the sand dune onto the parking lots through wind, through roots and other constraining measures. Another idea is a wood chip berm, pretty much along the same lines of the shrubbery. This sand wood chip berm would essentially be along this area here and would prevent sand from blowing from the beach from the north onto the sanctuary property. Note that both of these two solutions would take place on county land, so we would the NOAA would have to work with the county to accomplish this. Another solution is sand fencing. So sand fencing has been in place for approximately the past 15 years, but over that time, it's broken down. It's become, it hasn't really been doing its job. So investing some more money and repairing that sand fencing so it can return to its state of high operability can be very prudent to reducing the sand blowing out into the parking lots. And that sand fencing can either take place on NOAA property, this area right here, or on county property where there's already sand fencing in place. And finally, a wall, think like a cinder block wall, could be built on NOAA property that can reduce sand from blowing onto the parking lots. Granted, I found that often when this is done, the sand would blow into some other piece of land. So it's really just a temporary solution, not a long-lasting sustainable solution. Another problem that I found is drainage. So water is flowing from land east of the town on this high erosion area here, which is ranch land occupied by cows and goats. Due to the cows and the goats, they eat up a lot of the vegetation, which increases the possibility for erosion to occur. And when it rains, especially during the winter months, water will essentially flow down from the mountain, pick up a lot of sedimentation, and continue to flow straight towards the ocean. And I've seen pictures that'll essentially, actually I have this next picture, it'll flow down streets, which you can see right here. This is actually a street, as well as wetlands, which many of them have been built up. And when this happens, there's just so much water that will eventually get onto the road and flow south along this road right here, which is South Kihei Road, and eventually flow into the sanctuary, causing this effect right here, massive flooding of the sanctuary property. And approximately the water just stays here, and approximately 2 days later, the water will disappear. It'll drain slowly, leaving a very thin but dense, messy layer of mud over the sanctuary property and parking lot. This lack of ability for the water to drain is due to the bathtub effect around the sanctuary, due to the points of higher elevation surrounding the parking lot here. One solution I have that can combat this is adding a channel that can drain water from this bathtub area into a parkland directly south of the sanctuary, and from then into the ocean. Some other solutions possible solutions that can be installed include regrading the parking lot, so water naturally doesn't sit in the parking lot, it drains out of it, as well as installing permeable pavement, so water can actually drain through the parking lot. This is something that's been installed several places, both here in Hawaii and across the US continent, as well as installing a drainage flume highlighted in blue here that can essentially grab the water before it actually flows into the parking lot and direct it into the park and the ocean just south of the sanctuary. So the water just avoids the parking lots and the sanctuary grounds altogether. So overall, my main recommendations that I think that should really be looked at includes adding a drainage channel on the southern end of the parking lot. 2, insisting the County of Maui repair the sand dune fencing. 3, regrade the parking lot for improved drainage. And 4, add a drainage flume along the eastern and southern end of the sanctuary property to prevent water from ever getting into the sanctuary parking lot in the first place. So next steps for the sanctuary itself, I'd recommend that they hire a professional engineering firm to both gain a second opinion and to engage in the actual subcontracting and construction work that would need to take place to accomplish this. From an ethics point of view, I'm still an engineering student and am not allowed to make official professional recommendations at this time due to legal constraints. For me personally, I hope to continue my studies of environmental resiliency topics as a Fulbright scholar at the TU Delft Institute in the Netherlands, hopefully starting August 2024, after my 4th year in college. And after that, I plan to commission as an engineering officer in the US Space Force. So these are some of the sources I used to complete my project. I also like to thank my very great mentor, Patty Miller, both for providing me great mentorship during my time here, as well as just bringing me to Maui in the first place. It's been a great time here, as well as some other technical advisors and my funding sponsor, the Hollings Scholarship Program. So this concludes my presentation. I'll be happy to take any questions at this time.
Any questions for Derek?
Yes.
I don't see any in the room. Do we have any online?
None online.
Okay.
Great.
Thank you so much, Derek.
You bet. Have a nice day.
Great job. So next we have Simran Singh. She is a Pacific Young Scientist Opportunity Intern with PIP's Protected Species Division. Her mentor was Vikram Marana. The title of her talk is Transitioning the Hawaiian Monk Seal Biological Specimen Database.
Aloha everyone, my name is Simran Singh. I'm a PYSO 2023 intern and my project is transitioning the Hawaiian monk seal biological specimen database. Now a little background of the Hawaiian monk seal. So Ilio holo ika ua ua, which means quadruped running in the rough seas, can also mean the dog running in the rough seas. This is a Hawaiian name of the Hawaiian monk seal. Neomonachus struenslandi is a scientific name of Hawaiian monk seals originating from the German zoologist named Hugo Struensland, who visited the northwestern Hawaiian Islands called the Laysan Hawaiian Islands for research. Now, Hawaiian monk seals are the only endemic species in the Hawaiian archipelago, and they also happen to be extremely endangered, which is why they are protected under the the Endangered Species Act and the Marine Mammal Protection Act. So what are monk seals? What are Hawaiian monk seals in general? So monk seals are pinnipeds. They are fin-footed and they are true seals that do not obtain external ear flaps on the side of their heads, and they only reside in the Hawaiian archipelago. Now, comparing them to seal lions and other seals such as elephant seals, they are solitary animals. They do not congregate on beaches. Now, why are they called monk seals, which is a question that a lot of the Hawaiian monk seals get. So it is mainly because they have thick folds of blubber on the side of their neck, which resemble the folds of the robes on monks in the Buddhist and Hindu cultures. Now, they're also solitary animals, which is another reason. Now, as I mentioned, the Hawaiian monk seals are endangered. They have several threats to their population. A few threats are within the northwestern Hawaiian Islands, which consists of smaller islands and atolls, there's habitat loss and food limitation. Now, within the main Hawaiian Islands, disease and human interactions are a main threat. Some diseases are toxoplasmosis and morbility virus. Now, the population was 1,200 in 2007, but has thankfully increased to over 1,500 in 2023, thanks to HMSRP and NOAA and many other organizations for their conservation efforts. Now, it is split between 400 seals in the main Hawaiian Islands and over 1,200 seals within the northwestern Hawaiian Islands. HMSRP, the Hawaiian Monk Seal Research Program. Now, this program works diligently in order to conduct research and recovery activities to conserve our Hawaiian monk seal population. Now, some activities that they do are field camps for population assessment and recovery for the rehabilitation of our Hawaiian monk seals. Now, they also provide disentanglement and health interventions within the northwestern Hawaiian Islands and the main Hawaiian Islands. Regarding the veterinarian side, health and disease analysis are conducted. So the health and disease portion, there are necropsies that are conducted. Necropsies are autopsies of animals. So autopsies pretty much try to identify the cause of death of our animals to mitigate any threats to the population. Now, we also provide instrumentation where we attach cameras and satellite trackers to our Hawaiian monk seals in order to track their migration levels through different neighboring islands. Now, we also provide screening, which is situationally dependent because depending on their appearance and their behavior, we initially screen different monk seals for different types of viruses and bacteria. Now, we also collect samples and vaccinations. So the main disease we vaccinate our monk seals are for morbidity virus, which is a main cause of their death. Also, we provide abscess treatment where we see lumps on the side of their bodies and we provide incisions in order to drain the abscess. Now, my project is transitioning the Hawaiian monk seal biological specimen database. Everything I have explained on the other slide pertains to this database because everything is stored into SPA or this new database that I'm creating. Now, the problem is this database is called FreezerWorks. Now, FreezerWorks is a biological record keeping system. It is extremely problematic for us because it is not ideal for our program. FreezerWorks is exclusively built for hospitals and human patients. It is a medical record keeping system where you keep all of your medical records, samples, test results, and pretty much anything in general that relates to patients. Now, the main problem is that it is not user-friendly. We have to constantly walk around in eggshells just to keep up with the updates for FreezerWorks, and it does not pertain to our program because there are many features on FreezerWorks that does not allow us to input in information for monk seals. Now, this is the reason why we have not entered in any information within the past 3 years. We have been keeping in paper records for medical records of Hawaiian monk seals because there's extremely crucial problems relating to FreezerWorks. Now, here comes my role. My responsibilities is to utilize and troubleshoot FreezerWorks and transport all files from FreezerWorks onto this new customized Microsoft Access database. Now, this database is exclusively built for the Hawaiian monk seal population. Now, you might wonder, what is FreezerWorks? FreezerWorks is a fully configurable solution for sample management and sample collection and inventory. Now, it is not just for inventory, we also collect all of our medical history within this database. Now, we collect dry, wet, and frozen archives within our database, and it is organized into patients, samples, aliquots, tests, and test results. The main reason why we used FreezerWorks and began using it in the 1980s is because FreezerWorks obtains this barcoding system where we are able to attach barcodes onto our sample and easily scan this sample into our system. Now, since we're in modern day now, in 2023, we have now phones in order to barcode pretty much anything. So it is now not ideal for our program. We also collaborate with the NIST biorepository. They do not house any of our samples. We house all samples here at NOAA. So we just pretty much collaborate with the biorepository. Now, this is an overview of what FreezerWorks looks like. You can see it looks very similar to Excel, except we are able to input in all medical history and medical identifications of our monk seals. Now, if you look here in the middle portion of the slide, these are all of the bacteria, viruses, and diseases we screen our monk seals for. Now, we do not screen every single monk seal for all of these diseases. It is dependent on their appearance and behavior, so we screen different monk seals for different viruses and bacteria. Now, we store all of our test results in here. Now, FreezerWorks, although I mentioned it has a large list of problems, FreezerWorks is extremely crucial for our program, mainly because it is a biological record keeping system. Now, I'm sure we all know why biological records are extremely important. It is because it is the cornerstone of all research projects in the future for the conservation of Hawaiian monk seals. Now, we do house all annual 1,800 monk seal samples here. So annually we collect 1,800 monk seal samples, but we do obtain 80,000 Hawaiian monk seal samples in total here at NOAA. You cannot find them anywhere else. They're all housed in the 13 freezers we have here at NOAA. Now, if we lose any of these samples, it will be a complete detriment to our population for Hawaiian monk seals because In order to conserve, recover, and enhance their population, we need to conduct research from necropsies or any type of samples we are collecting from Hawaiian monk seals in order to mitigate threats and potential causes of death for our Hawaiian monk seals. Now, this is not the only reason why freezer works is important. Freezer works is crucial in order to obtain permits. Now, in order to conduct research and go out for stranding of our Hawaiian monk seals, we need to obtain permits. Now, this is why freezer works come into play, because in order to write permit reports, we need to submit the reports in order to get the permit in order to conduct our research. Now, freezer works exactly tells us how many samples we collected within the last permitting years in order to obtain a new permit. Now, the future of Hawaiian monk seal population research and conservation lies within this project and within this new database we're creating. Now, here's the solution. So FreezerWorks is the problem. The solution is this new customized Microsoft Access database. Now, we are developing this database solely for the Hawaiian monk seal population in order to store all of their medical history and all of their medical data. Now, a lot of people in the monk seal program talk about SPA, and they also talk about this new database. So SPA is our seal population assessment database, where we keep all of our life history of Hawaiian monk seals. And this new database contains all of the medical history. So these two databases need to talk to each other in order to create a full report of our Hawaiian monk seal. Now, we are initially trying to say goodbye to FreezerWorks. So I would like to thank my mentors, Claudia Cedillo, our veterinary technician and employee of the year, my other mentor, Vikram Kurana, our operations research analyst, my supervisor, Liz, the entirety of PSSP, Protective Species Division Support Program, Jessica, Hope Bronco, Claudia Cedillo, Vikram Kurana, Laura McHugh, our veterinarian, Michelle Barberi, and the entirety of the monk seal program, Brenda, Christy, Tracy, Mark, and Stacy. Here is my mentor and my neighbor, my references, and mahalo, thank you for your time. Oh, sorry, do you have any questions? Awesome, okay.
We have a comment that your presentation pictures are a lot cuter than the pictures in Derek's presentation. So great job.
All right, our last speaker of the day is, of the morning session, not of the day. You're not off that easy. Cesar Rodrigo Feliciano, and Cesar is a Pathways intern with NOAA's Weather Service. And this morning he's going to speak to us virtually on Caribbean tsunamis. Oh no, I'm so sorry. Vikram, are you here? There you go. Vikram, would you like to, I apologize. Vikram, would you like to say a few words about Simran? Yes, please, please. My apologies. In disguise.
Do I need a mic? Oh, thank you. Okay, I'm actually one of the two mentors for Simran, but the primary mentor is Claudia, and she's been working with her for the last 3 weeks while I was on leave. And she's on leave now, so I'm speaking on behalf of her. And this is what she said of Simran. Simran has been an enormous help with this project, and we couldn't be more impressed with how inquisitive she's been in learning about the team and all the work happening around the center. She's been wonderful to have around on the day-to-day and has been quick to volunteer her help to those in need. That curiosity combined with her hard work and kind spirit is sure to take her to great places and remind us of the great things to come from the next generation of scientists like herself. Okay, kudos to you, Simran. Thank you.
All right. So, yes, I agree. I would love to go back in time and be an intern again. It's so much fun. We are back to Cesar. Can you, there you are.
Yeah. I'm sharing my screen now.
You're speaking to us from Puerto Rico this morning, I believe. There we go.
Okay. Perfect. You see my screen?
Yeah.
Okay, perfect. And good morning. Yeah, it's morning there. My name is Cesar Rodriguez. I'm gonna be talking to you about the work I've been doing this past month with the International Tsunami Information Center in its Caribbean offices here in Puerto Rico. And well, I'll be talking about the Caribbean tsunamis assessments. There we go. A little bit of the agenda. I'll talk about the UNESCO IOC Tsunami Ready program a little bit. What I've been creating a Caribbean blue line, developing an evacuation map, and the next steps going forward with this projects. So the Tsunami Ready Recognition Program is an international community-based program developed by the Intergovernmental Oceanographic Commissions, IOC, of UNESCO. The main goal with this program is to improve the coastal community preparedness for tsunami in order to minimize the loss of life, livelihood, and property. This is achieved through a collaborative effort to meet a standard level of tsunami preparedness through the fulfillment of a set of established indicators, which are the indicators I have right here. Communities that meet all 12 of these indicators that include assessment, preparedness, and response will be recognized as tsunami ready. And in an effort to facilitate that through the Caribbean, I've been working to create a Caribbean blue line, and I've been also working with the evacuation map that I'll show you a little bit ahead. So creating a blue line for the Caribbean, a blue line is basically the easiest way to do it is just a line that when you cross it, you will be safe from tsunami hazard. And since a lot of the Caribbean islands do not have model data, or at least the one that I've been working on so far, I've been using the bathtub line approach that's been stipulated in the manuals and guide 82, while using the tsunami inundation zone for migration mapping guidelines by the National Tsunami Hazard Mitigation Program. Especially, I've been using a lot of the elevation, distance for shoreline, and the tectonic settings, because we're still in the initial phases of this project. And the first island that I was working on was the island of Guadalupe. So this island, due to its tectonic setting, for those that we don't know, the island of Guadalupe is on the northern section of the Lesser Antilles, and it's really close by to a subduction zone. So basing us from that tectonic setting, we decided that the best approach for the island is to do a 3-kilometer inland distance and a 30-meter elevation height for the blue line. And it's the things that I was doing. I first acquired a layer that was just the shape of the island. This is the island of Guadalupe. I also did a little bit of a zooming so it gets a little bit more clearer picture. Firstly, I did an inland buffer with those 3 kilometers. That will be what we are seeing right now. That's 3 kilometers from the distance from the shoreline. And then using global 30-meter DEM, since the island doesn't have a high resolution DEM, I had to use the SRTM 30-meter DEM, sorry, I've been saying DEM a lot, to get that 30-meter line. And extracting it, this is what I had. That green line would be the 30-meter elevation. Having those both, I was able to overlap them in order to create that blue line. And in order to do that, all I need is those 2 layers and the shapefile editing tools provided by QGIS to trace a line through the data that first reach inland from the coastline. So by that, I mean, if we take a look at the lowlands on the west, we can see that the 30-meter elevation came before the inland distance when we're taking consideration from the coastline to the first data reach. And we go to the highlands on the east, we can see that in some parts, the inland distance came before the 30-meter elevation. So I traced a line across that data, the first one that came, and then we created that blue line, which is what we're seeing right here. This is just a preliminary pass. There's still a lot of more work to be done with it. There's some data that we're missing, but this is how it's shaping out to be at the moment. There is a lot of issues that we've been facing, especially since it is a 30-meter resolution DEM. There's some occasions, as we can see in the screen, that the DEM, the 10-meter, 30, and up to this case, up to 60-meter, went up beyond the shoreline instead of going inland. So that's the main issue that we've been dealing with at the moment with the 30-meter resolution that we've been having. And aside from that, as I said before, I've also been working on developing an evacuation map for the island of Dominica, which is also found on the Lesser Antilles. We've been discussing with the community where they want the community, well, we're discussing with the community leaders where they want the community to be tsunami ready, be located, and they finalized the decision in the community of Portsmouth. So once we had the extent, we went to work. I utilized the Open Geographic Database OpenStreetMap, or OSM, to acquire the base layers. That was just the Caribbean Sea, the land. I also applied a 5-meter DEM hillshade to give it a little bit of texture so it doesn't look that plain. And using, again, the OSM tool, I was able to extract the infrastructure from the island, the roads, the buildings, and then having that data, it was compared with satellite imagery to determine what was up to date and what wasn't up to date. So if we take a look at the center top part of the island of the map, there's some buildings right there. That's a hotel that was not part of that initial OSM layer. I had to manually draw that in so it was more up to date. Once that was done, I took those aside and I worked with modeled data from the University of Singapore in Costa Rica. And they did 20 different sources modeled and then compiled it in one composite, which is what we're seeing. This would be the inundation layer. And going from that, I was able to create a smoother version of it to create an inundation extent. And once I had that, I again put all the roads and buildings, and using, again, shape validating tools, I went following the guidelines that the evacuation zone had to be extended to the next road possible. It couldn't cross property in half, and it should also be continuous through the shoreline. And once that was done, and then was an open discussion with the community, basing themselves of what inundation they've seen and what they haven't seen, we finalized what was the first draft for this evacuation map. And this is how it's looking at the moment. Again, this is just the first draft. We're still in open communication with them to see what they want to add, what they want to remove, where it's going to be assembly points, all that. But this is basically where we're at the moment. And going forward with both these projects, firstly for the development of the Caribbean Blue Line, the data needs to be validated because this is just basing us in distance and elevation. So we need more data to have it as accurate as possible. We want communication with the different countries to see what data, storm surge, inundations, all that kind of data they have, to add it to that layer, have it more accurate. And we want to also assist the countries that are seeking to be tsunami ready, but do not have modeling. And the end goal of this is to repeat the methodology with the remaining countries in the Caribbean and adjacent regions to establish a region-wide blue line, incorporating both non-modeled and modeled, where available, inundation extents. And with the evacuation map, we're going to continue supporting the community of Portsmouth in the process of creating this evacuation map through open discussion with them, provide QGIS assistance and feedback for the evacuation map, and assisting it in getting that tsunami ready recognition. And well, yeah, that's what I've been up to this past month out here in the International Tsunami Information Center in the Caribbean office. Thank you.
Thank you. Any questions for Cesar? Yes. Here.
So I'm probably also the mentor, but hold on. Hi, I'm also the mentor, but the question I had for Cesar is if he can let us know what he thought is the most challenging part of Tsunami Ready that he's involved in implementing, because he's involved in not only Guadeloupe and Dominica, but he's seen all the other countries of the Caribbean. Cesar?
Yeah, that's a great question. Definitely the hardest part for me has been getting the data, because this is really important work. We're trying to save lives, trying to save properties, livelihood. So I want to get this as accurate as possible, just so that I can help save as many lives as possible and not cause more panic than necessary. Because since, again, the blue lines, like it's very general, I want to get it as accurate as possible so that people that aren't necessarily in danger, that I'm putting them in the danger zone at the moment, I don't want them to panic for no reason whatsoever. So definitely getting the data for implemented and having it as accurate as possible has been the difficult part for me. But I'm definitely working towards getting more data, getting it more accurate, and trying to establish those communications with the different countries to see what they can provide us to get that more accurate.
Wonderful. Cesar, would you be able to unshare your screen so we can see you? I wanted to say thank you, gracias, for your presentation. It's just really cool having a hybrid platform like this so that we can have people from other parts of the country share. So we really appreciate your willingness to present virtually. Thank you.
No, yeah, thank you for the opportunity. I'm glad to be here. I'm really excited for the work that I've been doing, and I'm looking forward to continue my work through my remaining time with Pathways and international and with ETIC.
So I had a question. I was curious if you were able to make recommendations to maybe the city and county or state agencies there about biological remediation, building mangroves, not having beach hardening, or not building the roads too close to the shoreline to mitigate the level of inundation should a tsunami occur?
Me personally, no, because I've been mostly working with the GIS aspect of this project. So I haven't gotten to directly talk with different countries or different officials. But those are interesting points, and I'll definitely pass them along to my superiors so I take it into consideration. Thank you.
Any additional questions? Yes.
Stealing Caitlin's job. I was wondering if you're using ArcGIS, if you maybe are going to integrate this into maybe an app on a phone that people can use so people can know where and when they need to get to higher ground in case of a warning or an alert. Maybe do you plan on doing that?
Me personally, no, but that is a goal that the project itself has. We want to do, I don't know if we're going to do an app per se, but definitely they are visualizing a web page that people can go see and know where they're at, or they have to evacuate and where not.
Any additional questions? All right, Kristen, would you like to come up and say a few words? Oh, you're not. I'm sorry. I have Krista Van Hilbrand is his mentor. Yep, right there.
There's also a quick comment that says, congratulations. Great job.
Thank you so much.
Congratulations. My name is Laura Kong. I'm actually the director of ITIC. We're based here, but we also have an office in the Caribbean. So Cesar is in the Caribbean. He works directly with Krista Van Hilbrand Andrade, but she's actually at a meeting in Germany. So she sends her congratulations, but she asked me to say a few words. So just generally, Cesar was actually a student intern before the Pathways opportunity came. He's got a working in the background in geology at the University of Puerto Rico in Mayagüez. So kind of an excellent person by which to not only talk about the science of tsunamis and the science of around the Caribbean, because he's not working in Puerto Rico, he's working in Guadeloupe and Dominica and St. Lucia. But when the Pathways program was able to hire him for the summer, immediately what Krista did was find a project that would have an immediate impact. So he was able to learn from some other interns who are there. One person whose name is Desiree. She was actually at Pathways last year. So working together, what they've done is create a methodology, as he said, where there is no good DEM, where the bathymetry is just not as good as we'd like, but create a methodology that can be used not only in Guadeloupe, and they've done it in Jamaica, but throughout the Caribbean. So kind of help us along with this goal, which is actually at a global level to get 100% of all communities at risk for tsunamis prepared and resilient by 2030. So there are something like 49 countries and territories in the Caribbean, and I think they worked on maybe about, I don't know, 5 or 6 or 7. So really helpful and kind of moving us along for, I think, as he said, kind of looking at the global effort. So thank you, Cesar.
Thank you again for your time and attention this morning. We're going to go on a short break. So please plan to be back in your seats by 10:40. You'll hear a little bell at the 2-minute warning. So just kind of be alert for that. So we want to start the next session promptly. The coffee and snacks in the back are provided by Halea. If you want to buy a mug, see Don Kobayashi. They are, you're welcome to use them. We encourage that because we want to not have waste, but please do leave them if you don't plan to buy one. So, but please help yourself and thank you so much.
She's a Hollings intern. She's working with the NOS, the National Ocean Service Hawaiian Islands Humpback Whale National Marine Sanctuary. And the title of her talk is Coral Reef Education and Outreach for Hawaiian Audiences. So I'll go ahead and turn it over. Actually, I'll turn it over virtually to Laura.
Yes, hello everyone. Allow me a moment to set up my screen. All right, can everyone see my screen?
Yes, we can.
Okay, perfect. Hi everyone, my name is Laura Jones, and I spent my summer internship at the Hawaiian Islands Humpback Whale National Marine Sanctuary doing coral reef education and outreach for Hawaiian audiences. So we'll go over the background and the objectives of my internship and then what I actually did this summer. A little bit of background about coral reefs. As we all know, coral reefs are amongst the most important ecosystems in the world. They provide economic and environmental services. They are biodiversity hotspots, and they also have cultural significance, but they're under anthropogenic stressors that are threatening their existence. A lot of these are due to climate change and are the result of ocean acidification. And then there's also effects due to pollution, runoff, and coastal development. Hawaii's coral reefs are particularly important because they are central to the state's culture, livelihood, and economy. They're home to over 60% of the United States coral reefs, and their total economic value is estimated to be at over $33 billion. Not only this, but they're essential to local culture, which I found out this summer. For example, in the Kumulipo, the Hawaiian creation chant, it tells us that coral is one of the first things that is created, which eventually gave rise to more complex organisms and eventually humans. So this teaches us that we're all connected and it's all thanks to coral. But unfortunately, they are prone to sediment and nutrient pollution from sewage and stormwater runoff. There's been bleaching events in the past due to extreme weather events and El Nino events. So they're extremely important to protect. A little bit of background about me. In elementary school, I was obsessed with school and becoming a teacher. The picture on the left is me in second grade dressed up as my teacher for Halloween. But as I got older, I kind of forgot about that love for education, and I focused my interest more on marine biology. Growing up in San Diego, I was always around the ocean and loved learning about it. So it eventually became my major at the University of Miami, where I found a particular interest and passion in the field of coral reef. I've been involved in coral reef research. So when I was looking for an internship, I was looking for something that combined my early love for education, my love for coral reefs and conservation. So my internship was spent at the Hawaiian Islands Humpback Whale National Marine Sanctuary, and overall the overarching goal was to target conservation, but I wanted to do this at the root. So this was done by creating a coral reef education unit. This would be used both at the National Marine Sanctuary and for distribution to local teachers. And then also I got experience teaching coral reef lessons to student groups here on site. And then lastly, part of my internship was participating in and creating materials for outreach events with an emphasis again on coral reefs. So the Hawaiian Islands Humpback Whale National Marine Sanctuary focuses a lot on whales and whales disentanglement, which is during whale season in the winter, but year round, and especially during the summer, they focus a lot of their efforts on education and outreach, which is what I was doing this summer. So in order to complete this, I created a coral reef curriculum. There are 5 lessons in the curriculum. They are interactive and multimedia, so they're not just PowerPoint presentations, and they really target kids in building a foundation of science knowledge, science literacy. Lessons 1 through 4 are for 3rd through 5th grade level students. This is because at the Hawaiian Islands Humpback Whale National Marine Sanctuary, that's the age group of a lot of the summer groups and the school groups that come in. And then lesson 5 is for pre-K through 2nd graders, because those also tend to show up at the sanctuary. Each lesson is 45 to 60 minutes, and lessons 1 through 3 can be taught independently or consecutively. And then lessons 4 and 5 are if the teacher or instructor only has 1 lesson, 1 shot, 1 time period to teach about coral reefs, and they consolidate those lessons and the topics from the previous 3 mentioned. So walking through the lessons I created, the first one is about corals and coral reefs. Teaches coral reef biology and anatomy and lays the foundation for more complex ideas about these ecosystems. The picture on the left is an activity I helped refine here at the sanctuary where students made an edible coral polyp. So I walked them through the anatomy and the structure of the polyp, and they got to create their own model of it, and they really liked it because they got to eat it, which was fun. I also included several videos from NOAA and other sources that enhance the learning. Lesson 2 is about who lives in a coral reef. So this is more coral reef ecology. I created a scavenger hunt here that was a murder mystery food chain, looking at some of the goals for Hawaiian Common Core education. For 3rd through 5th graders, they highlight the importance of understanding what a food chain and food web is. And so I decided to make this funner and interactive by making a scavenger hunt. Lesson 3 is about coral reef conservation. So first I talk about some of the threats to coral reefs, but I really wanted to have a positive spin on it because I could talk forever about how reefs are damaged and the negatives of coral reefs. But in order to inspire conservation from a young age and motivate people, especially young students, to learn about coral reefs, I really wanted to have a positive lasting impact on the lesson. And so I have an activity where the students model what would happen to coral reef skeletons under ocean acidification. And then I also designed coloring pages that students could take home, put on their fridge to remind them what they can do to actively participate in the protection of coral reefs. And then, like I said, for those consolidated lessons, those are combining topics from the previous 3 lessons I created. And then the only difference is that lesson 4 is for younger students and lesson 5 is for older students. Here are some pictures of me at the Hawaiian Islands Humpback Whale National Marine Sanctuary, teaching the younger students about who lives in a coral reef and who doesn't belong in a coral reef. It was really fun, and it was really fun to engage with them and listen to them and their ideas about what coral reefs are. And then now shifting towards what I did for outreach. First, I created materials. So this were brochures, pamphlets, handouts, coloring pages, things like that. An area that I really want to highlight in terms of coral reef conservation were how sunscreens affect our oceans. As we know, in Hawaii, there are bans on certain chemicals for the protection of coral reefs. But I found quite early here that a lot of people don't know why the coral reefs are, or the sunscreens are affecting coral reefs. So I wanted to bridge that gap. And then also I helped out with creating social media posts for the Facebook and Twitter of the Hawaiian Islands Humpback Whale National Marine Sanctuary to increase engagement and community awareness of what we have going on here at the sanctuary and coral reefs. And then also I participated in events. On the left is a booth that I held at the Maui Ocean Center very recently for their coral spawning event, which was cool. And then on the right is a picture of me and my coworker Derek at the sanctuary doing with a huge inflatable whale that we have and kind of helping kids through that and teaching them about certain parts of the whale. So to summarize, as we know, coral reefs are important but threatened. Hawaii's coral reefs are very crucial to the state's economy, culture, and livelihood, but through education and outreach, we can target conservation in order to preserve them. And then here's just by the numbers some of the results that came out of this internship, such as the number of summer groups I hosted, the handouts designed, posts I curated. And then on the right is kind of the folder to visualize the, like I said, the products of my internship. And then the NOAA goals that I hit were the engagement enterprise and healthy oceans. The next steps for this internship or future internships would be to continue to spread the coral reef curriculum to Hawaiian teachers and measure impact. And then also I think it would be a really good idea to create an Instagram to increase the social media engagement and the marketing of the Hawaiian Islands Humpback Whale National Marine Sanctuary. Thank you to my mentor, Patty Miller, who provided me with great guidance throughout this internship, as well as staff members, Michelle Graham and Daryl Peck for their support. And then thank you to the NOAA Ernest F. Hollings Scholarship Program as a funding sponsor. Any questions?
Thank you, Laura. That's terrific. Any questions? Maybe time for one question. One question from the audience or online.
Hi, oh, sorry, that's really loud. Thank you for your presentation. You're very lucky to work with Patty. She's the best. I had a question. You might have mentioned this, but what did you use to build those coral polyps with the kids? Because I thought that was a really fun activity that I might want to do myself.
Yeah, Patty is the best. I second that. But the materials were a graham cracker for the substrate, a marshmallow for the soft body, icing to represent the calcium carbonate structure, little pretzels for the tentacles, and green sprinkles for the zooxanthellae.
Very cool.
One more question. Thank you so much. That was awesome, Laura. I am interested in using some of the materials that you produced, some of the outreach materials. Is there a way that folks can access those?
Yeah, definitely. In the next week, in my final week here, I'm going to be working a lot with my mentor, Patty, to really finalize how she's going to distribute my materials. But you're welcome to email me and I can share with you the Google Drive folder that I created with all the materials.
Awesome. Thank you so much. Great job.
Thank you.
Thanks so much, Laura. Appreciate you. All right, next on tap, we have Trevor Nishida. He has a PYSO, Pacific Islands Young Scientist Opportunity. Did I get that right? Close? All right. Internship. He's working through the Science Center with the Ecosystem Services Division. Oh, sorry. And his title of this talk is Aquatic Analysis Adventure, Exploring Biodiversity Patterns as a Hawaiian Reef Ecosystem Using eDNA. And his mentor is John Whitney. So I'll turn it over to Trevor.
Thank you, Leon. Hello, everyone. I'm Trevor. I'm a student at the University of Hawaii at Hilo studying marine science. Today I was going to give you a talk on my PYSO internship and kind of what I've done over the past almost 2.5 months that I've been here. So first, I just wanted to give a big thank you to my advisors, John Whitney, Jamie Gov, and Andrea Schmidt. And I'd also like to give a thank you to Justin Suka, who helped me with some of the data analysis portions of my project. So let's first start by defining what eDNA is. For those of you who don't know, eDNA is short for environmental DNA, and it can be described as DNA captured or sampled from the environment without direct contact with the organisms. So this can come from the soil, snow, air, but in our case, it came from the water. So in the marine environment, eDNA can come from a plethora of different sources. So for example, we have dead and decomposing organisms, habitat builders, such as our coral reefs and emu, benthic inverts, such as sea cucumbers, from waste or excrement products from these organisms, as well as their mucus, scales, and skin cells. eDNA can also enter the water through sloppy feeding by our top predators, such as sharks and jacks. And we can also pick up eDNA by looking at the microorganisms in the water column and our planktonic species. So all of these are ways that the genetic material from these organisms can enter the water column and be sampled by us, the scientists. So a few pros and cons to eDNA sampling. So it is non-invasive in that we don't have to have direct contact with organisms, which may stress them out. And as we saw earlier with the shark bycatch presentation, can cause mortality issues. It also allows us to have higher resolution for cryptic species detections, meaning that we can find species that we may not have otherwise been able to detect. eDNA typically has higher detection rates than traditional surveys using visual censuses, as well as providing us with the opportunity to kind of remotely sample these environments without having to deploy divers to sample directly in the water. We can just take water samples from boats and use that instead of processing samples that were collected through scuba or snorkel methods. Most importantly, however, eDNA provides us a way to sample across all trophic levels, meaning that we can get representation from microbes, from the level of microorganisms all the way up to our top predators, such as those sharks. Some cons to eDNA is that the DNA is subject to degradation in the environment. This is especially true with marine environments where DNA is actively decaying over time at a much quicker rate in saltwater environments than in freshwater ones, as well as being exposed to high levels of UV radiation from the sun. Another issue with eDNA is we cannot really pinpoint the spatial location that the DNA was collected from. So this can lead to things such as pelagic species being detected in our coral reef environments, or terrestrial species being detected due to runoff from the land. eDNA cannot derive biomass data because we are not seeing the fish ourselves, so we can't get an estimate for how big these fish are or how many they were. There's also the issue of cross-contamination, such as I said, the water from our terrestrial environments washing out and us getting terrestrial detections, as well as in the lab, it might become, it may be really easy for us humans to leave some of our DNA in the detections. We are also very limited by our reference databases, and so with our public, with the public databases, they are relatively poor quality and do not really have localizations. So for a place such as Hawaii, where we have many different endemic species, it might be difficult to distinguish them in using these large-scale public databases. Another might might not have there might not be enough distinguishing features between two similar species, meaning that we can't get a species level identification on some of these breeds. So for the first part of my internship, I had to create a regional invertebrate list, as there was no easily accessible one for the economic exclusion or economic exclusivity zone of Hawaii. The first is the invertebrate checklist, which is looking on the side here. This is pretty, and the last part will be computer.
It takes. Sorry to interrupt.
Hello, hello. Okay, we're back. So yeah, as I was saying, this data was pretty difficult to work with. As you can see, it was just a really big text file listing from phylum all the way down to species level identification. And a lot of these identifications were either outdated or not updated to the newest taxonomic classification. Some of the other databases that were used to source this list were community source. Along with that comes with a little bit of messiness with incorrect spellings of species, as well as incorrect identifications, as they were done by pretty much citizen scientists. Another interesting thing to note about these other databases is that they included preserved specimen data, which are things that are located in, or say, the Bishop Museum as a preserved specimen, but might not be something we expect to see here in the in situ marine fauna. So I created a couple data tools that would take a list like the one you saw on the previous slide and create something that looks like this. These tools use the World Registry of Marine Species, or WHRMS, to fix the taxonomy of species and make sure that we have a relatively updated version of the taxonomic classification for these individuals. They also compare entries across databases, these databases being the Ocean Biodiversity Information System, or OBIS, as well as lifewatch.be, which is a database which had entries from a bunch of different other ones, including GBIF and AFIA. We just wanted to compare between databases to make sure we don't have any duplicates. And then it also removed the preserved species entries. So those are the things preserved in museums, as well as correcting any incorrect spelling using the WHRMS official naming and deriving the full taxonomic tree from kingdom to species level. So this list was then used with data collected last April on the SE 2202 cruise. This cruise took place across the west coast of the Big Island of Hawaii. This coastline is roughly 225 kilometers long, and about 113 survey sites were stratified into 12 different levels using environmental factors such as the substrate type as well as structural complexity of the strata. At 81 of the sites, eDNA sampling occurred, and the goal was to survey the entire reef ecosystem biodiversity using eDNA combined with traditional visual census methods and compare these results across ecological gradients. So to accomplish this, at each site, a number of different surveys were completed, the first of which being the structure from motion photogrammetry surveys done by the Mega Lab located at UH-Hilo. These surveys produced a 3D model of the coral reef, allowing us to have coral, algae, and benthic invert identification, as well as extracting some benthic characteristics such as rugosity and percent coral cover. Underwater visual fish surveys were done by DAR, which allowed us to have fish identification, as well as abundance and biomass estimation from their visual data. Finally, eDNA water sampling was done by NOAA, in which we can get fish, coral, algae, plankton, and invert identification, so pretty much spanning the entire trophic, all trophic levels. For the eDNA sampling, approximately 6 4-liter containers of water were sampled at each site using the setup seen here with a pump and a sort of cube tainer. These were then put on ice until they were transferred to the ship, where they could be filtered using this setup here. These filters were then sent off to another lab to be identified for eDNA, or to be eDNA meta-barcoded. So in this process, the eDNA, or the DNA data, is amplified using PCR, which is polymerase chain reaction amplification. If that sounds familiar, it was a way to get tested for COVID when the pandemic was first hitting the world. So after that, we looked at a certain set of markers, which is an area in the DNA to identify different kinds of species. So we used a total of 6 markers, the first of which being CL1, which is across metazoans, 16S and 12S to identify fish, 18S for eukaryotes, and then ITS2 for coral, and 23S for algae. So the result, some preliminary results here. These are reef fish species detections across both eDNA and the visual census survey methods. So a total number of 307 unique species were identified. Here you can see the list of the top 10 fish species detections across both of the survey methods. Something to note here is that a large portion of the top 10 were surgeonfish. Surgeonfish are important as they contribute to the herbivory on a reef, preventing the shift from a coral-dominated reef to an algal-dominated one, which would not be as ideal for our reef fishes. So looking at fish exclusive to the visual survey, something to note here is Thalassoma duperi, or the saddle wrasse. This fish was one of those instances where there's not enough distinction between species to get a species level identification through eDNA. And this results in something that we can only identify the genus, which is why it shows up here exclusive to the visual survey. Another thing to note is about half of the top 10 fall under 10 detections across all 81 sites. Looking at fish exclusive to the eDNA survey, these top 2 fish here are schooling fish, which would, a large schooling fish, which would represent a large portion of the eDNA data as they are leaving large traces of their DNA in the water column, as well as cryptic species such as our soldierfish, eels, and blennies and gobies. So the next steps in my internship would be to explore the relationships between biodiversity and environmental drivers. So here is just a non-metric dimensional scaling plot comparing depth gradients to the species richness, and also to dive deeper into heavily targeted fish such as Uku, Omilu, and Roy using methods like these, as well as boosted regression trees. Finally, to explore human driver relationships such as population density, nutrients, and fishing pressure. I will now take any questions, and you can scan the QR code to see my references.
Any questions for Trevor?
Sure.
Trevor, great presentation. At what depth were the samples taken where you did the eDNA analysis? And did you notice there's a certain depth by which if you're up on the surface, the dilution is so much that you can't get a good sample?
So I actually wasn't directly involved with the sampling process, but this plot kind of is displaying that. So samples were taken in ranges from less than 5 meters all the way to depths that were greater than 15 meters. Those depths greater than 15 meters, I think our maximum depth was in the 20-meter range. But as you can see here, or what's displayed on this plot is basically that there was no distinct variation between these different depth groups for sampling. And this is, yeah, so there's no distinct variation between depth groups when looking at it from a, I guess, species richness point of view.
Do you have time for one more question? Or? I do want to turn it over to John Whitney, who can say a few words about Trevor.
Great job, Trevor. Trevor's been incredible to work with. It's being like working with a PhD student and an advanced one at that. He has enough material to do an hour seminar. This was such a small piece. He's creative and productive and brilliant. It's really been a lot of fun. We presented him with challenges that we have in dealing with this eDNA data, which is all very new. There's lots of informatics, and every challenge that we put in front of him, he solved it quickly. Within the first 2 days, we knocked the first challenge out. Within the first 3 weeks, he created the most comprehensive list of all the marine flora and fauna of Hawaii, which hadn't existed yet, and made it reproducible and shareable, and created some permanent resources that will benefit our projects for years to come. So it's incredible to see him work. We're gonna grab him and keep him for as long as we can. Hopefully get him a graduate student fellowship or something, however long we can keep him around. Yeah, it's been really fun working with you. Thanks, John.
So our next speaker is Ashley Hawking. She's a Hollings scholar working for both my office, OCAO, as well as the Ecosystem Sciences Division in the Science Center. And she's going to be doing a speed talk entitled The Reefs Revealed, Embarking on a 3D Journey of the Blob's Lasting Impact. So I'll turn it over to Ashley.
Thank you, Leon, for that introduction. So yeah, hi everybody. My project's called Reefs Revealed, Embarking on a 3D Journey of the Blob's Lasting Impact. So really quickly, a little bit about me. My name is Ashley. I have a background in environmental science and studio art. And I've always really enjoyed combining these passions to help bridge the gap between environmental science and the public through accessible and engaging formats. And so in line with this, I actually have a 3-minute speed presentation, but that's because I have homework for you at the end. So we're gonna jump right in, talk a little bit about the blob. And so what is this? No, I am not talking about your classic sci-fi thriller from the 70s. Instead, I am talking today about a massive marine heat wave. So this occurred about from 2014 to 2016, had a huge impact in the North Pacific Ocean, but stretched along this range. And we saw unprecedented sea surface temperatures, and with that, unexpected impacts on our marine ecosystems. One of those being major coral reef bleaching and mortality. And of course, as we know, global warming exacerbates natural fluctuations in our environment. And with this, we're going to see pretty big events. And I'm not just talking blob-like in size, I'm talking also about severity and frequency. So marine heat waves have become more and more common and have had drastic impacts like this. And so there's a need for efficient data collection and monitoring. So what's NOAA up to? As Trevor briefly mentioned, we've been implementing structure from motion technology. So what this looks like is as divers are swimming underwater, they're taking pictures from every angle in order to stitch these into 3D models to create digitized maps of coral reefs, which are really awesome for analyzing all sorts of different things and also tracking change over time. But what's cool about these is they provide new perspectives without needing to be underwater. And not only this is really helpful for scientists, this has been a great educational tool. So for the summer, I've been working on mapping or linking ecological data with a time series of the Oluwalu Reef and the impacts that the heat stress events in 2014 and 2015 had in coral at the Oluwalu Reef, and connecting these to educational standards to try to create an educational video that can be implemented in classrooms in a 3-part series. And the story of the Oluwalu Reef during the same time that the blob was occurring is a story of mortality in some ways. The video explores a little cauliflower coral going through a classic, bleaching event and dying. But it's also a story of differential resilience, where we see 2 lobe corals that were living side by side. On your left, we see one that was experiencing bleaching and partial mortality, but on your right, there's a coral that was healthy and was able to resist and recover. And this is really important for scientists and for us to understand when we think about coral restoration. When we look at coral that are already adapted or have strategies to resist the heat stress. And the video also is a story of recovery. So there was 2 rice corals. One was bleaching in 2015 next to a mostly healthy one, excuse me. And in 2016, we saw that it actually recovered while there were some parts that died, and they fused together. So here are some other scenes from the educational video, just to get you hooked. And then if you're interested, I have the QR code. This is one of the later drafts. There's still some tweaking before the final symposium, but you can find this. It's about 9 minutes at the moment. That's why I had to do speed talk. This is your homework. But also for homework, I really recommend that you let yourself fall in love with coral reefs. I mean, this project taught me so much, and I'm so grateful for my mentors, Leon and Tom. Thank you to Corinne, who taught me how to use VisCore, which was really hard, and do those fly-throughs. Thank you to Laura, who taught me about educational standards and connecting the science to education, and how can we have something engaging for students. Thank you for Kimberly Martin, who came before me as a scholar and prepared a lot of this information that's in the video. But yeah, let yourself fall in love with coral. It's really easy for us to pay attention to the big charismatic megafauna, and there's nothing wrong with that. But coral is not just economically important. I mean, it has huge cultural significance. It's important for protecting our islands by dissipating wave energy and storm surges, and it hosts over 1/4 of marine life. So thank you, everybody, and please enjoy the video. If you watch it, let me know. I appreciate any feedback.
Thanks, Ashley.
Thank you, Leon.
Yeah. So I do want to, just say a few words about Ashley in terms of, I think the theme of resilience really fits well with what she had to encounter this summer. I mean, just everything in terms of, dealing with on the programming side, dealing with the educational side, having to do with both scientists and educators, and finding the right balance between the two is definitely a challenge. So I think you really exhibited resilience in an amazing way. So I do want to thank her work there. And just to point out, this video, we'll be using this from here on out in terms of educational programming for people coming to the IRC. So it's a great part that it's just not a summer program, but something that we'll see implemented here at the IRC as well as in classrooms as well too. So I just want to acknowledge all the tremendous work that you did, and thank you for always, and she'd always bring up great questions and would always be asking for feedback. So that's a great skill to have, to be able to know how to ask for feedback and incorporate that into the future iterations. So great job, Ashley.
Thank you, Zion. Thank you, everyone.
All right, our next speaker is Haley Durbin. She's a Hollings scholar working with, I guess, protected species and PIRO. And the title of her talk is Catching Seals Red-flippered, Training AI to Detect Hawaiian monk seal Interactions with Marine Aquaculture. And her mentor is Stacy, who'll give a few words afterwards. So I'll turn it over to Haley.
Okay. Can you hear me? I think so. Okay. Hi, I'm Haley. I am a student at the University of South Carolina studying marine science. And my project title is right there. So I'm just going to dive in. So the main question that I'm going to kind of go through today is, or my project is seeking to answer, is digital monitoring an effective approach for monitoring interactions between protected species and aquaculture operations? Digital monitoring is a term that's used to define like any kind of use of technology tools, like cameras, sensors, satellite tags, things like that, to monitor or observe, collect data of interactions in like water or the environment, anything like that. So there are a couple different kinds of aquaculture facilities that are out there. There's land-based systems, recycling systems that are controlled indoors, and water-based systems in things like ponds or inshore, offshore systems. But what we're going to talk about is open water offshore systems at a specific aquaculture facility in, an offshore system. And this is kind of the top half of the aquaculture net pens. And so just keep this like cone structure in mind as I go to this 2D diagram. So this is the top half, and the bottom half is almost exactly like the clip that you saw before, but flipped upside down. And throughout the day, this is raised to kind of prevent biofouling on the top half of the pen. So, and then lowered and completely submerged in the other parts of the day. So we have a couple cameras that are set up that were put there a year ago-ish. And these are just some of the points of view that we have from the cameras. So this is the first one that's looking down at the bottom of the net pen where it kind of connects to the structure that connects it to the sleeve floor. There's also a camera that faces the outer rim of the fishing net. And this is kind of what it looks like when the fishing net is completely submerged. And this is what it looks like when it's kind of halfway above water. So just for some reference points of view. So what happens when you put a huge fish net in the middle of the ocean filled with a bunch of fish is you get predators that are attracted to it. We had some chocolate in the pantry the other day, and I think I saw more people in there than I've ever seen. That's kind of like what this is like. So we've got some sharks, dolphins, Hawaiian monk seals, and some of these species are endangered and protected under different acts. But this is kind of what the visits are in the film that I've surveyed and gone through. And as you can see, there's almost a visit every hour. So they're pretty much always there, the predators. So what we're focused on is Hawaiian monk seals. And there's a few reasons why we're kind of focused on this for the specific aquaculture facility. One reason is that they're endemic to Hawaii and very endangered, as you kind of saw from Simran's presentation earlier. And they're protected under a bunch of acts. And recently, or I guess in 2017, there was a fatality within one of the fish nets. So that was kind of a big deal and had to be resolved pretty quickly. So since then, these fish pens have been monitored by the farm staff and by NOAA, kind of in conjunction, trying to solve some of these problems, which is why this is here. But so these are the human observations from the farm workers and how many there have been over the last like 3.5 years. And as you can see, just kind of for fun, the interactions are kind of increasing, at least the observations of the interactions are increasing. And I don't really have an answer for this, but other than the fact that we know that the populations of monk seals are increasing with efforts to restore their populations, and that is my best hypothesis of why they're increasing. So there's a couple different behaviors that we see at these fish pens, and it's just swimming, interactions with other species, as you can see these pictures here, and approaching falling vessels, fishing attempts, and just pulling fish out of the fish pens. So with our trial run of digital monitoring at the fish pens, there are cameras set up at different angles that we saw before. These cameras are running for 4 days just because of battery life, and they run during the daylight for 12 hours. So for storage purposes, they're then clicked into 10-minute segments because storing a video footage that's 12 hours long is really impractical and would be a very large file. So with this digital trial run, we want to know, are the cameras capturing seal interactions, and is AI a possible way to kind of go through all this data in a more efficient way? The answer is yes, we are seeing interactions on the cameras that are co-aligning with what the farm workers are seeing in person. And as you can see here, we know that they are getting fish from, it's kind of orienting weird, but if you flip it upside down, that's kind of this is the bottom of the fish pen, and he's pulling fish out of this gap in the bottom, and that's kind of already been dealt with the farm, but it's just a confirmation of that's how they're getting fish out of the fish pens. And here you can see, oh no, that he's pulling it out kind of just on the side of the net. And we think these fish are dead, and the mortalities that are within the fish pens. So this is kind of a future problem maybe to work out, but and how to mitigate and deter the seals from pulling out the mortalities from the fish pens. And they are, the farm workers are going and removing those mortalities every day by their protocol. So like there's still some issues there even when they're removing it every day. So how do we analyze all of this footage if they are running constantly or more often than just 4 days? Yeah, so if human analysis is an option, which is what I kind of did with all of the footage that we have. And that took a really long time, and it's very large amounts of data and large amounts of video watching, which is just not super feasible if you have that much data. So is an AI model an option? And the answer is yes, but it takes time to train. But once it's trained, it's less human power. So to kind of go through all this. So how I trained this AI model to kind of identify and detect when seals are in the frame. We have about 760 videos total, and those are like the 10-minute clips. And then I clip those down to about 30 to a minute and a half segments, and use those to train and run through the model after annotating them, kind of identifying when sharks are in the film, monk seals, fish, whatever kind of predators that came through the camera frame. So that worked. There's still some future things to be done with that, but overall we know that it's an option moving forward. So what are some of the pros and cons between digital monitoring versus just like human monitoring with the divers and the just people on the boats, the workers at the farm? So digital monitoring has continuous monitoring, non-intrusive, unbiased. But it also has very few points of view. You only see what's in that camera frame, whether that's the bottom of the net pen or kind of facing outwards, and the monk seals might be anywhere around the net, but you only see what the camera can see. But with human observations, you have a lot more points of view. You have the divers, the boat workers, whoever is out there, but just a lot of monitored interactions and things like that. So there's some problems with comparing and combining this data, which I was trying to do just to see what if any of the monk seals I spotted on the cameras were coinciding with the human observations. And so this is kind of the graph that I got from that. And as you can see, because I count visits, so every time the seal enters the screen, I count that as 1. And if it leaves and comes back in the same day, it counts as 2 visits. So obviously there's not 77 monk seals in like 1 day of filming. So that's kind of like a weird way to compare, but at least we know that there are like coinciding on the same day. So there's like a monk seal being seen when the human observers are also seeing monk seals. So takeaways are the methods by which we steal the fish from the pens are identified and modifications can be made. They've already been making modifications over the past couple of years. So this is just kind of enforcing those and allowing NOAA and the fish farm workers to work together and move forward with whatever modifications they want to make. And now we know that AI can detect monk seals in videos. So this is a possible method of analyzing these videos moving forward. And the question, another question moving forward for people to answer, is it worth the cost of making this, figuring out ways to store that footage? It's a lot of footage. And so if you have that much data in one space, there's like a lot of storage issues and things like that. And server like availability to run the AI on that much footage is also kind of a problem. So is that worth it? But here's my thank yous to all of the people that have helped me. Stacy, my mentor, and Tori, Heather, and Savannah have been a super big help with all of these things, and just the whole PIRO team has been great, and PIFSC. But anyway, it's been fun to work for both sides. But thank you. That's it.
Any question or two for Haley? Guess I'll go first, Kate, and then
Thanks. That was really great. I'm curious why it's so bad for the monk seals to eat a dead fish. Is because they need to keep an eye on mortality fluctuations within the pens, or?
Well, it's not like necessarily them eating the dead fish. I don't think that's the problem. It's just their interactions with it are causing like behavior differences in their normal like call out behaviors, and also just if they're hanging around there, they're just getting like a ton of fish and getting fat and happy and hanging around there. So it's not like what their normal interactions would be in life. And I mean, there have been some mortality, like a fatality in it, and another one got caught, but they ended up like making some modifications where it was able to be released and didn't have any damages. So like, there are just some other risks that are involved when they visit.
Kate, you had a question?
That was a great, that was a great presentation. I was really curious. If I recall, there's a small resident monk seal population on the Big Island, and I was wondering if the AI could be set up such that you could actually detect which individual species are frequenting the net pens more often, like in just seeing maybe which ones, and if there's any characteristics or reasons why those seals are going to the net pens and not others.
I'm honestly not sure. I mean, the AI has a hard time detecting, like, it's hard for me to detect which seals are there and which ones aren't, so I don't know how well that it would do with detecting, like, which seals, which species are there. So that might be kind of difficult with AI at least, but maybe. I don't know.
You taking it back, right? I know. So did you want this one?
Yeah. Question online. Okay. Well, I'm going to turn it over to Stacy. You can come up and.
I just want to say a huge thank you and congratulations to Haley on this project. This fish pen project started years ago, and so Haley really kind of like swooped in and helped us do so much, kind of like tightening it up, getting things analyzed that, you know, she spoke from very viscerally true experience about some of the difficulties with the data storage and data movement and processing. But yeah, I just can't appreciate enough how much Haley has just like jumped in wholeheartedly to this. And we've put her through like so many different situations with being co-mentored across PIFSC and PIRO, and, you know, kind of in like her first week here, we're like, hey, do you want to learn like a completely new AI program that you've never seen? And also the Section 7 guys really want to talk to you about the implications of your project. You know, so she's just really taken all of that in stride and been so just cheerful and welcoming of input along both sides, and just done a really fantastic job. And we are absolutely now able to use this new tool that she's developed for us. Awesome job. Thank you.
Terrific. So our next speaker is Jade Winkler-Barden, who's a Holling scholar working for PIRO, and the title of her talk is Understanding Carbon Emissions of PIR Longline Fleets, a Literature Review. So I'll turn it over to Jade.
All right. Hi, my name's Jade, and this is going to be a literature review of understanding carbon emissions of PIR longline fleets. And just so you know, PIR, if nobody knows, is Pacific Island region. And so longline fishing has been a main source of food and revenue for over 100 years. And if you're not sure, longline is a fishing method that is used with a very long line, and you have multiple hooks attached to it. And so that's how it catches pelagic species. And so today's long line has 150 vessels, and it has two segments where it catches big-eye tuna and swordfish, where fishing vessels are in the main ports are in Honolulu and American Samoa, and these vessels can be out for a month up to multiple months. And so the main focus of this work is to essentially understand and calculate the carbon and greenhouse gas emissions from our Pacific Island region fishing fleets, starting with longline fishing and so this map shows pretty much like the jurisdiction of the Hawaiian Islands longline fishing fleets. And so fisheries of the Regional Management Organization includes the Western Central Pacific Commissions in the blue shaded area, and the Inter-American Tropical Tuna Commissions in the lighter blue area bounded by the gray dashed line. And so that's tropical tuna. And the red zones are exclusion areas. This project was started as an action item of the Pacific Islands Region Action Plan of Climate Sciences, specifically to estimate the carbon emissions associated with activities under NOAA fisheries jurisdiction in the PIR, where possible potential emissions can be reduced. And so these are some, we have NEPA, which is the National Environmental Policy Act, and then we have the PIRAP, which is the Pacific Islands Region Action Plan. And so in 2023, guidance was issued under a National Environmental Policy Act that federal agencies assess potential effects of both greenhouse gases, emissions, and any greenhouse gas reductions to better help decision processes. And so what I've been doing is I've been reviewing 17 journal articles and technical reports. They've covered 5 different fisheries of longline, purseine, pole line, aquaculture, mariculture. And I've compiled 20 different equations. And so this is pretty much like assessing like fuel use intensity, which is FUI, and that is expressed as liters of fuel consumption per amounts fish landed. And pretty much it's going to be a lot of variables, and so a lot of it like compiles of vessel size, the type of engine, the amount of time each vessel is out, and the landings, which are the catches. And so FUI helps compare fuel use between fisheries and species and can be converted into estimate of greenhouse gas emissions in kilograms per carbon dioxide when fish are landed. And so in the next couple of slides will pretty much be like graphs and figures of what I reviewed and pretty much explaining what they all compile of. There'll also be a lot of limitations and variables within every single graph and chart. And so this graph is a global perspective of the fuel use and greenhouse gas emissions of world fisheries. This is from Parker in 2018. And so this is pretty much like the shows like worldly, and A shows the global landings in thousands, which China, Asia, United States have a lot of those, has the most out of the whole entire world. The greenhouse gas emissions for labeled as B, China again, and Asia has the most. For world map C, the greenhouse gas intensity is going to be pretty much in Australia region and a little bit of Europe. And for D, it's going to be the percentage of food production emissions in the fishery. And so that shows mostly a little bit of North America and South America, little tidbits of that. And so a lot of variables used in this was the vessel engine size, the number of fishing in days per year. It also found the median fuel use intensity for the global fishery record since 1990, which came to be 639 liters per ton. And so this article discussed emissions by the category of fish targeted, which was small fish, crustaceans, and large pelagics, which is a limitation in here because, you know, that's not really our main focus. All right. And so this literature review from Tymers and Parker in 2012 shows a table of Pacific longlines, which is highlighted in yellow. And so this is essentially to like review how compare the 3 different fishing techniques and how much fuel is burned. And it also shows like the specific estimates found in fisheries targeting skipjack, yellowfin, and what were the most like efficient, well, the least efficient fisheries. We're targeting albacore tuna and bluefin tuna. And so a couple limitations here is that the study relied on the voluntary participation of the industry, and the data limited could allocate a time and efforts required to gather the data and complete the return surveys. Was most successful at collecting data from purse seine and the targeting skipjack and yellowfin species. All right. And so in this most recent study, they pretty much focused on United States tuna fishing fleets with mostly, you know, long line and trawl. And so it was they were evaluating the sustainability of different tuna fleets, including long line, and taking into account of the climate impacts and the interactions of protected species with sea turtles. And so this pretty much like broke down the type of gear and targeted species that they assessed. And so as you can see, the red is trawl, light blue is purse seine, dark blue surface, and gray is longline. And so you could see that trawl has the most impact than any of the other fishing. And so in 2015, this is a graph from 2015, and it was noted that additional upstream process associated with fishing included vessel construction and maintenance, gear manufacturing, bait provision, and consumed pretty much more emissions. And so this is pretty much to show how like not only fishing can contribute to the greenhouse gas, is that it could be the processing, it can be through transport, And so it's important to consider not only fishing activities, but also, on the fishing fleets. And so shown here, the Pacific has a lot less than the Atlantic and the Indian Oceans. That's also because the Pacific has smaller contributions than the Atlantic and the Indian Oceans. And so here are some ways to reduce emissions from longline fishing fleets. And so it's important to note that officials are trying to hold future warning to only 1.5 degrees Fahrenheit. So they have to take into consideration not only fishing, but transport, fuel use, processing. And so everyone's looking to green power, and green power, they're looking for wind, sail, possibly adding sails onto these fishing vessels, using hydrogen as a source of fuel, and even going 5 knots slower when they're pulling the line. And so next steps into my work is to finalize literature reviews. Then we are going to determine how we can calculate the emissions of longline fishing fleets and possibly gather any necessary data for this work. And then lastly is to expand from Hawaii to the American Samoa, Guam, and the Commonwealth of Northern Mariana Islands. And to look for smaller fishing fleets too, because this is pretty big. And we're continuing to look for more information. So if anybody has any stuff on that, that'd be greatly appreciated. And then I would just like to thank my mentors, Kate Taylor and Lee Hing Chan. They've given me a great opportunity, and I'm just so appreciative for it. I also like to thank the IRC and the NOAA Hollings Preparation Program. I've just enjoyed my time here and my work. Thank you.
Thanks, Jade. Time for maybe one or two questions, Jade.
Check, check, check. Thanks, Jade. I can appreciate a fellow literature review project. There's one graph you had that showed the same fishery, the same species over time, and there was quite a drastic change in emissions. I wonder what you attribute that to.
Not quite sure. Not sure if the literature review really assessed on that. Can you tell me what graph it was again? I'm so sorry.
Yeah, right there.
This one? Yeah, I'm not too sure. I mean, this was focused. It was really hard because this was mostly just the United States. It was nothing really on Hawaii. So yeah, I'm not sure. Maybe they're officials just trying to crack down on limitations and, how many days people can go out on, or the different types of fishing material used.
Any other questions? Maybe one more?
We have one question from the chat.
Oh,
or from the online presentation.
You want to read that, or?
Elcall, feel free to unmute yourself.
Oh, Alex, yeah, go for it.
Thanks. Mic coming in. Audio check.
Yeah, we can hear you.
Yes, go ahead.
Cool. Awesome. Great presentation. The International Fisheries Division, obviously very interested in this sort of work. One question and then one comment. Question is there any sense for the Pacific having that lower intensity footprint? My guess would be that that's just related to the dominance of Persane in the Pacific relative to the Atlantic and Indian. So I guess the question is that true, or are there other explanations for that difference? And then just finally, if there was interest in taking a closer look at the Pacific fisheries managed by the WCPFC, I would be very happy to collaborate in helping acquire the data to create this analysis for the WCPFC, and that would certainly be a paper of high interest to the Commission, just understanding the greenhouse gas emissions of the fishery managed within the WCPFC. Thank you.
Thank you.
So it was hard to hear some of it, so I don't know if there's a chat. We can probably follow up with you, Alex, on that question. I don't know if did you get some of it, or
I got Persane and comparing it.
I had a hard time hearing it.
So sorry.
But maybe we can answer it offline if that's okay, Alex. All right. Well, I do want to call up Jade's mentor, Kate, who will give some kind words here. And just a reminder, they're reminding me that we should stand near the podium next to the mic so people can hear online. So honestly,
Great job, Jade. I want to acknowledge that Jade probably has the distinction of being the person who traveled the farthest to be here for the symposium today, because she flew in from the East Coast yesterday just to make it here for this symposium. And she is also part of the Hollings prep program. So she's about 3 weeks into her project, and we just kind of threw this at her. We kind of, both HIHWNMS and I, we kind of gave her this topic, and we said, oh, by the way, there's a symposium in 3 weeks. Do you want to present at it? And she's like, sure, like this will be a good experience. And it was a really great learning experience and a good preparation for the symposium that they have at the end of the Hollings prep internship program. So for being 3 weeks in comparison to some people who have been here for 2 months, I'm really proud of the work she's done already. And I hope that next year she will apply to the Hollings Scholar Program and is getting inspired by a lot of the great talks that we saw here today. So thank you.
how are we doing last? All right, our next presenter is Helena Sigos. She is a LaPenta scholar working with the National Weather Service Forecasting Office. And the title of her talk is TC Verification Trends Related to Evolution of Meteorological Tools. So I'll let you. Thanks, Helena.
Hello.
Is that on? Oh, perfect. Hi, everybody. My name is Helena. Thank you for the introduction. I'm a rising junior at Cornell University studying atmospheric science. And today, this is my presentation on the Pacific tropical cyclone verification trends and their relation to certain meteorological tools and their evolution through time. So my two motivating questions for this research are, which meteorological tool has the greatest utility when forecasting tropical cyclones in the northeastern and central Pacific region? And then following this, how does the evolution of that tool compare to trends found in the 2-day and 5-day outlooks of tropical cyclone verifications throughout time? So the methods we can see here. So the NHC separates and grades their forecasts based on two categories, intensity and track of hurricanes, or sorry, tropical cyclones, either. You can see here, so I created a list of tools and made sure that the obs that they collected are related to intensity and track. And I went through and slowly eliminated some of these tools based on their spatial and temporal availability and the history of their use throughout the Pacific region. on these TCs, and also judging their ability to be used on every TC in the region, so more spatial. And then for the verification trends, I basically took data from the National Hurricane Center's Central Pacific Hurricane Center's Hurricane Verification Archive. It was able to grab air data and skill data from the baseline, basically plot those, see if there's any trends, and I ran some statistical tests on those. And also I wanted to make note that for the trends, I stuck between the parameters from, or spatial temporal parameters from 2001 to 2022, because in 2001, that's when the first 5-day outlook was issued. So my results, so here you can see a list of the tools that I had created. And I kind of went through each one and decided based on the parameters that I was judging them by, that satellites are the best tools for spatial and temporal regions or reasons to be used in this region. For example, so the two satellites that are most oftentimes used by the National Weather Service and the CPHC in this case were the Polar Orbiting Satellite, which is also known as the NOAA satellites, which are identified in blue up here, and the geostationary satellites, or GOES satellites. And so basically I created this timeline by hand, it took forever, of every single satellite from the period that was active from the period of 2001 to 2022, basically saying when they were launched, when they weren't working. So these areas in like dark green and dark blue are when they were like out of commission, and when they end were when they were permanently out of commission and set up for storage. It took me a while. There were lots of resources that kind of argue back and forth between them when they were launched and when they were online and whatnot. And then this highlighted pink region at the very end, I'll talk more about that later, but that is when the first advanced baseline imager was implemented in GO 17 in 2019. And then I kind of have two other pictures down here that depict maybe why we can't really use some of these other tools. For example, with the buoys, we have this chart, which basically is a map of every single certified NOAA buoy that is available to take OBS. And we can see that climatologically, if you overlay hurricane tracks and cyclogenesis in that area of that red region, that's where the highest amount of cyclogenesis occurs. And you can see that there are no buoys in that region. So that's probably not going to be very useful when trying to predict these storms. So you can't really use those. And then as far as the this picture, I pulled it up actually 2 days ago. You can see that this is actually a map of wave heights. That bright yellow blob right there is Tropical Storm Calvin. And you can kind of see that these are boats and they're all around the yellow blob. So not really going through the hurricane to collect OBS for us, because that would be bad. But yeah, and they have ASOS stations mounted on those. So they're like little mini weather stations, and they will give us OBS, but obviously they can't go take OBS through the hurricane. And moving on, kind of our results in terms, since we established that satellites are the best tools to use for this region, I kind of took more of the data from the verification archive from the NACH CPHC and was able to create a bar graph of forecast skill on that side and error on this side. So for track, we judge it based off of nautical miles, and here we can see that the green is the median of the error for nautical miles. You can see that as you go on throughout time, the box plots get smaller, which means that our data is being more clustered and there's less outliers and there's less variability for error, which means that our forecasts are getting better, which is good. And then for our 120-hour outlook, same story. You still have that decrease in the median, less outliers here, although there are some more, and I'll talk about that in a bit. But it's kind of a less steep of a curve. And then we go on this side, similar story. We have our our skill increasing throughout time, and less steep with the 120-hour, because obviously that is a larger temporal parameter. And you are going to have greater accuracy, obviously, with 48-hour rather than the 120-hour. And then similar story for the intensity, except this time we're judging it based on for error. On knots, so you can either overestimate or underestimate your wind speed. So that is why we're trending towards zero, like zero air wind speed. For the 48-hour, obviously trending more so towards that median of zero air. over time compared to the 120 hours. It's a little bit more slow to get to the that sweet spot, but you are still getting there over time. And then very similar story with results for intensity, except you see that negative dip right there, which there's some reasons for that. More, I guess, variability in those seasons with ENSO. I'm also going to be talking about that in a little bit. But you see a less steep of an increase in skill over time. But as I was saying earlier with the advanced baseline imager, my project is not finished yet. I still have about 2 more weeks. I plan to make a graph that will predict points of error and skill on a plot for the next 10 years for the advanced baseline imager and how that has had implications on forecasting. And as far as how we are going to relate it to ENSO, I plan on making trying to do some correlation and statistics relating the El Nino and La Nina of each season and how that correlates to data points throughout time and seeing if there's any cyclical, I guess, similarities in the data points. That is all I have for you today. Thank you so much for watching my presentation. I hope you all have a great day.
Lana, maybe one question for Lana. Ian.
Hey, just one question on the intensity part and with the ABI.
Yes.
What are your thoughts so far on how that's going to improve the intensity forecasting?
I kind of foresee it. Obviously, you're still going to have those outliers according if what I said was true about the ENSO and how there are so many factors that can have implications on the exact, I guess, accuracy of the predictions for each season. But overall, I see a very large decrease in error and increase in skill over time to the advanced baseline imager, just because that instrumentation gives us clear pictures, clear imagery. It introduces new bands of just wavelengths in which we could see the weather and the the hurricanes on satellite, and just better imagery. And it's also taking pictures on a smaller temporal timescale, so more pictures more often, so we can make little like movies like of the past and the going to the present to try and help with predictions. But yes, I do see a great decrease over time in error. Thank you so much.
All right, let's give Helena.
The final speaker for this morning is Jessica. She's a Hollings scholar working with the Protected Species Division. Her title is Honu in Motion, Studying Abundance and Trends of Green Sea Turtles in Papahanaumokuakea Marine National Monument. Turn it over to Jessica.
All right, thank you for that introduction. So my name is Jessica Lipschultz, and my project this summer was studying abundance and movement trends of Hawaiian green sea turtles in the Papahanaumokuakea Marine National Park. So first, for a little bit of background on the monument itself, it is almost 583,000 square miles of protected area. So for scale, this monument is approximately half the length of the continental United States. So it is a huge area in the central North Pacific Ocean, home to many threatened and endangered species, one of which being the Hawaiian green sea turtle, or Honu. So Honu in this region of the world do a really unique behavior called basking, which is when they pull themselves up onto the beach and they'll just chill there for a while for reasons that we still don't really know. And they only do this in 3 regions of the world, here in Hawaii, the Galapagos, and some regions of Australia. And another really important part of the PMM is that it houses their primary nesting site, which is a small atoll called Lalo, located here. And we have a lot of really awesome data from Lalo over the years. There's been consistent annual basking and nesting surveys all the way from 1970. So from this information, we know that it supports approximately 96% of Honu nesting in this region. So on average, we see about 400 nesting females a year. So we have a lot of great information from Lalo, but we don't really know much about the northern part of the monument, which is approximately half of their habitat. So I call this region the North Hawaiian Islands, or the NHI, and it includes Kamolai, Kapo, Wanabai, Midway, and Hawaii. So this is where I came in. So my job was to look at all the data that we have for the North Hawaiian Islands and see what I could make from that data. So it was a really big task to start, and I started off with two main questions. Number one was, what is the state of the available data? What's even out there? What do we have? And next is, what data analysis is possible? So what are the most consistent metrics that we have been taking over the last 50-ish years? So that started a really intense review of every single NOAA data sheet, tech report, and literature that I could find. And I found a lot of interesting stuff, but the main question that nobody really seemed to have an answer for is how many honu are even up there. So I completed an abundance analysis. So to do this, I gathered up all the basking surveys that we've done. And a basking survey is basically when a member of our field team walks around the entire perimeter of an island and just counts how many honu are there. So to make these trend lines, I found the maximum census count for basking survey in a single season for each year at each island. So this is the first time that we're seeing any kind of basking abundance trends for the North Hawaiian Islands. And then I was curious about the total abundance of basking turtles in the northern part of Hawaii. So I found the average abundance of basking turtles at each of the islands, summed them together, and I got 52. So we are seeing on average each year 52 basking honu in the northern part of the monument. So the main takeaway from this graph, other than that number, is that Kapo, Wanabai, and Midway seem to be able to host the most amount of basking honu out of the entire northern islands. So then we are thinking about things that could be affecting abundance, and we're thinking about resource availability. And resource availability can then affect the size of our honu. So then we were wondering if the size was varied between the islands. So then I completed a size analysis, and I looked at all of our curved carapace length measurements that we have for all of our turtles. So curved carapace length is a measure of the natural curve of the turtle shell all the way from the tip, or all the way from the head to the tip. So it is demonstrated by my colleague Brittany here. And the main takeaway from these graphs, we don't need to go into the specifics here, but the main takeaways is that there is no significant difference in honu size between the islands. Next, there are significant differences between the size of the females and the males at all of the islands. So females are larger than males at all of our northern islands. This is consistent with what we know about sea-free sea turtle populations in other parts of the world. But then we were curious, you're looking at our size analyses, and we were curious about how this compares to their primary nesting site of Lalo. So I grouped all of the northern Hawaiian Islands together, and I compared them to Lalo. So the main thing that I want you guys to take away from this graph is looking at these two top lines here. The top line is our Lalo females, and then the black line is our North Hawaiian Island females. And so the difference between these two lines is actually statistically significant, meaning that our Lalo females are larger than our North Hawaiian Island females. So there are a couple of reasons why this could be. Number one, Lalo, again, primary nesting site, is attracting all of our really healthy, ready to migrate, ready to nest females that could be contributing to why we're seeing larger size. The other reason is we do believe that most of our nesting females at Lalo are coming from our main Hawaiian Islands, which likely have better foraging grounds and more resources than those in the North Hawaiian Islands. So that could also be affecting their size. So after we looked at size, we went back to our abundance analyses, and we were again thinking about the things that could be affecting these trend lines. And again, we used 50 years of data to make these lines, which is obviously a very, very long time to be collecting data, and inconsistencies are bound to happen. So we were wondering if that was really affecting our abundance lines. So we decided to look if there was a possible survey bias. So to answer this question, let's zoom in on our midway data for a second. So what you're seeing here is a graph of our average sightings of honu seen throughout the year. The box you're seeing represents the average start and the end times for an average field season at Midway. So main takeaway from here is that our average field season is actually missing a lot of our peak basking that is happening later in the fall. So because we're missing our peak basking times, that means that we're getting inaccurate abundance estimates for Midway. So we looked at this for all of our islands, and you can just quickly glance at all of these, and you can tell that our peak basking times are outside our average survey times. So we are consistently missing peak basking. Then we zoomed in a little bit more and looked at are basking survey timings of the day. Again, we are missing peak basking times. Again, this is seen throughout the entire Northern Hawaiian Islands. So this could definitely be an implication for future surveys. We can try to align this value with peak basking so that we can get more accurate estimates for our abundance trends. But what does that mean for this graph here? So since we're missing our peak basking times, that means that it is probable that these numbers are actually underestimate of the amount of honu that are basking in the Northern Islands. So then, last step here, we were thinking about abundance trends and how many honu are at each of the islands, and we're wondering if honu were moving between the Northern Hawaiian Islands. So we do know that our honu in our main Hawaiian Islands have very high island fidelity, meaning that they have their resident island, that's their home, they stick to it, and they just go to wallow for their reproductive migrations. But we don't really know what's going on with the Northern Hawaiian Islands. So to examine this question, I looked at all of our tagging information from the entire monument for all honu for all years. And you can already see from this map here that the most honu are moving between wallow and our other northern islands. So as it turns out, there are 140 honu that have been sighted at multiple islands within the monument. Of those 140 honu, 139 of them are seen at only 2 islands. Only one turtle in the entire history of the program has ever been seen at 3. So this means that they are probably sticking to that trend of high island fidelity like we're seeing in the main Hawaiian Islands. And since most island connections are between Lalo and the other northern islands, it's probably reproductive migrations that we're seeing. It's not confirmed, but it is a clue as to how they're moving within the monument. So of these 140 honu, 15 of them were seen at multiple islands within the same year. And this is cool because that means that we have really interesting timing data for when they're moving between these islands. And so for most of these honu, they were seen at Lalo during nesting season, which is usually peaking in June and July. And they were seen at the other northern islands during other parts of the year. So again, this is our first clue as to how honu are moving within the Northern Hawaiian Islands. And it seems like they are following the trend of the main Hawaiian Island honu and going to Lalo for reproductive migrations. So if you want to remember a couple things from this presentation, I want you to remember these things. Number one, that Monavai, Kappa, and Midway are hosting the most honu out of the islands of the northern PMM. And next, that with more tags, increased effort in better aligned survey times, we can better understand resites and abundance trends. So now you might be wondering, what are the next steps for this project? So number one, we can definitely explore the possibility of modifying our survey times to better align with our peak basking times and get better abundance estimates. Second, we can increase effort on getting tagging information. So tagging has had a lot of increased efforts since 2017. We are putting a lot more permanent tags on our honu, which is going to be really important for when we are trying to track our resites. And lastly, I know from experience that you can definitely go into some very deep rabbit holes looking at this data, and continuing to explore the data sources for more insights would be a very interesting way to spend some time. So all of the information that I presented here today is going to be very important for NOAA when it is expected to present on upcoming status assessments for honu populations for the Endangered Species Act and International Union for Conservation of Nature. So big mahalo to the entire MTBAP team here at NOAA and the Hollings program staff. Could not have done without you guys. That's all I got for you guys. Any questions? Yeah.
Ashley, a question?
First of all, incredible job. That was such a thorough presentation, and I know that that data was like a monster, and so congratulations. In the beginning, you mentioned that they're not confirmed reasons for why turtles bask, and I'm wondering if that's something you've thought about and maybe want to explore more, or if you had any clues.
Yeah, it is definitely a very interesting question that I would definitely be interested in exploring. So there are a couple hypotheses. So one of the main ones is thermoregulation. So they're hauling up on the beach to they're reptiles, so they are cold-blooded. And another hypothesis is to help with digestion. And it could even be a social behavior, which could be cool, but it's definitely a very interesting field of study for the future. Yeah.
Any other questions? So,
Just I'm wondering in the graph in the top right, what explains the abundance decline in around 2000, and then what explains the abundance rise around 2014?
Yeah, great question. So that is definitely another future area of study that we could explore. So one possible thing that could be affecting abundances in the Northern Islands is weather data. So one other possible area of study is to look at fixing weather events like El Nino and see if that's lining up with our abundance trends. But obviously this is like a long-term decline, so it would be a little bit different than that. But yeah, it's a great question.
okay, I think we're okay. Let's wrap it up. But let me have Mary Lou. Is Mary Lou here? Oh, pardon me.
Hey, great. Thank you so much. That was such a great talk. I was really excited in the back there. I don't know if you could see I was doing some fist bump. But Jess, when she came to us in the beginning of the summer, when we wrote the proposal for this internship, we didn't really know what she would be able to get away with because of the state that the data was in. And I mean, literally her first week here, I was handing her like dusty piles of paper and like to see what she could do with it. And so she went through it all, that lit review, trying to compile all the data and then see if any of it even connected. We struggled, I know, with do we have enough size data? Do we have abundance data? We thought about nesting. And so all of the things that she's been able to do this summer were just so impressive. I know every time she had a chance to update the program or talk to someone in the program about what she was working on, the room just like lit up, and there was so much enthusiasm behind this work because it has been such a data gap for us. We have to focus on Lalo every year. It's our primary nesting site, and so this has really been this kind of back burner project that we've been really excited about for a long time, and so stoked that she could come and take it on. And we've been really impressed with all the coding and the troubleshooting you've been doing, well beyond my expertise, and going around and really making the most of the internship, talking to so many people in the building and the different management side, and helping us with necropsies, which are terrible. So thank you for that.
Very smelly.
They're smelly. They're very smelly. And we're happy to reward you with a tagging day on the North Shore next week. So thank you so much. I can't emphasize enough how important these data will be and her results will be for the status assessments that we're working on, and just knowing what's going on across the whole range of the population. And so we're very excited about staying in touch and hoping to see more of you in the future, and we'll have to get you a basking project. So thank you. Great job.
Thank you.
Okay, sounds good. So yeah, we are a little behind schedule. Apologize for that. But we are resuming at 1 o'clock. So if folks can be back here at 1, we do have our kind of interns meet scientists thing up on the 3rd floor on the lanai. So if people want to head up to the lanai with their lunches, we'll be there. But just be back here at 1 o'clock or a little bit before for our 3rd session.
And there will be cookies. There will be cookies after lunch. So come back.
Take a look, see if that's to your.
Okay, cool. So thank you for the introduction, and thank you all for coming here today. Today I would like to talk about sail drones and the acoustic data that we've been looking at. Just get set up here real quick. Oh, cool. So over the past month, I have been a part of a collaborative mission between 2 NOAA line offices. The Pacific Marine Environmental Laboratory, located at the Western Regional Center in Seattle, Washington, and the Pacific Island Fisheries Science Center, located here at the Inoue Regional Center in Honolulu. And the overall objectives of this mission is to study ocean atmospheric interactions and environmental effects on pelagic organisms in the central equatorial Pacific Ocean. So this is a very vast and remote region. So it's very hard to collect surface data in these regions because they're so remote. And especially for like research cruises, voyages can be very lengthy and super expensive, like $35,000 per day operating costs, as an example. So one approach that we could take to reduce this cost would be to use uncrewed surface vehicles, such as a sail drone, as you see here. So the primary objective of my internship and my role in this mission is to evaluate the use of sail drones to collect backscatter data on pelagic organisms for ecosystem-based fisheries management. So with that, we can improve models in these data-poor regions. So some of you might not be familiar with sail drones, so I would like to quickly talk about them. Sail drones are one type of uncrewed surface vehicle that are manufactured and deployed by Sail Drone, which is a private company based in California. They run on a business model where they sell the data to customers such as NOAA rather than selling the individual vehicles. And I think that goes for a rate of like $2,500 per day, if anyone's interested. For this mission, we are using the smallest sail drone model that they have, which is the Explorer. For reference, there's a person there. These things are about 23 feet long or 7 meters. They run on wind-powered propulsion and have solar-powered sensors. So it's 100% renewable energy. Each sail drone is equipped with an array of sensors. For the Sail Drone Explorer, it's equipped with 3 acoustic sensors, 4 atmospheric sensors, and 5 oceanic sensors. These are the methods of our mission. On June 22nd, 2023, this year, we deployed 3 Sail Drone uncrewed surface vehicles from Oahu. As you can see there, this is before the deployment. They're in port right there, or it's got shipped over. We deployed Sail Drone 33, 79, and 30. Each sail drone is equipped with a standard suit of oceanic and atmospheric sensors, but only one of them has the acoustic payload. The sail drones have sailed to the remote central equatorial Pacific since the 22nd of June. And I guess right now they're near the Kiribati exclusive economic zone, highlighted in red there. And this is a 120-day mission, so it should be done in October, I believe. We are utilizing an adaptive sampling approach to study air-sea interactions, surface CO2 fluxes, ocean currents, and backscatter data, which is what I would like to talk about next. If you're not familiar with backscatter data, backscatter is essentially the reflection of sound waves off surfaces within the water column. So this could be fish, crustaceans, whales if there are any, submarines, whatever. And we measure this in kilohertz. We typically use frequencies of 18 to 200 kilohertz frequencies. And essentially, the lower the frequency we use, the deeper in the water column the sound waves can penetrate. So if this podium was the water column, 200 frequencies would only go like to this depth, but like 38, for example, would go all the way down there. So each organism has a frequency-specific backscatter. For example, if we're looking at micronecton fish, which I'll explain later, they dominate the 38 kilohertz frequency. and this can be influenced by their body shape, size, orientation in the water column, and whether they have a gas bladder or not. For this mission, we are looking at 38 and 200 kilohertz frequencies down to 1,000 meters in depth. To measure backscatter, we use a device called an echo sounder. It's composed of two main components, the transceiver and the transducer. Then the transducer sends acoustic frequencies down into the water column, as you can see here with this graphic, and it also picks up the acoustic frequencies that are scattered back to it. And what this gives us is an echogram that looks like this. And this is actually a screenshot from the sail drone mission portal that we use to view live data and updates from our deployed sail drones. The x-axis represents time. This specific example is from July 3rd to July 8th at 5-minute intervals. This echogram also shows day and night intervals, which will be important here in just a second. The y-axis represents depth. We're looking at a depth between 5 and 1,005 meters. The pixels on this echogram represent the density of organisms, and how we view that is by measuring it in decibels, which is essentially proportional to density. The red, orange, and yellow pixels that you see on the echogram correlate or correspond to a high density of organisms. The green can be interpreted as like a moderate density, and the blue and gray can be interpreted as background noise or a very low density of organisms. What we could actually see from this echogram are the daily vertical migrations of micronectonic organisms. For example, near the surface or the epipelagic zone, we could see these clusters of green, yellow, red, orange pixels right there. And that is part of their daily vertical migration at night, where they feed near the surface. And during the day, we could see that they've migrated back down to about like, I'd say like 300 to 400 meters down. And we could see those clusters there in the mesopelagic zone. So micronecton are essentially organisms between 2 and 20 centimeters in length. They account for a substantial amount of biomass in the pelagic regions, and their daily vertical migrations are important for nutrient cycling and carbon transport between the mesopelagic depths, between 1,000 and 200 meters, and the epipelagic depths, the upper 200 meters. So the next steps in my project, I've only been working on this for like a month, but once we get the data back in October after the mission is over, we plan to process acoustic data to assess relative biomass and to assess how environmental variables may influence micronecton distribution, biomass, and composition. And with that, I'd like to acknowledge my mentors and everyone else who has helped me through the project. And does anyone have any questions?
All right, we have a question, and friendly reminder to wait for the microphone before you ask your question. Great job.
Hi. Oh, okay. Well, I'll just talk loudly. I was wondering, so you said that it is solar powered. I'm wondering if there's any kind of backup energy source that's on the sail drone and what that backup source of energy is. Great presentation, by the way.
As far as I know, the sail drone model that we're using, it's the Explorer model. It just runs on renewable energy. I'm not sure if there are any backup sources, but what I've read online is the larger models, they contain like, I guess, a diesel powered backup source, something like that, I think.
Any more questions?
Microphone appears to not be working.
Great job, Quinn. I was curious about the sampling frequency. You said it was samples every 5 minutes. Is that
Yeah.
the data that you have access to via the satellite, or is that the sampling frequency?
So that's, I guess, because on the mission portal, we could see like, or customize the graph to different frequencies, like 1 minute, 5 minute, 30 minutes. So that's just, I guess the one that, I mean, it displays the low quality data that we have. So if that answers your question.
It's that button.
Any questions online? All right, thank you, Quentin. Great job.
Thank you.
Testing, testing.
While we're switching out speakers here, just wanted to let the interns know if you could please stick around. After the last presentation, we're going to do a group photo in the atrium. So don't run off. Thank you. All right, next up we have Brooke, a Hollings scholar, and she's going to talk about media communications, developing crisis communication plans for severe weather events. Go ahead.
Thank you. So, hi, my name is Brooke Weiser. I'm a rising senior at the University of Miami studying meteorology, and today I'll be presenting media communications, developing crisis communication plans for severe weather events. So this summer, I had the opportunity to work at the National Weather Service office over on the campus of UH-Manoa, the Honolulu Forecast Office, and I worked with John Bravender, the warning coordination meteorologist there. So the overall goal of this project was to develop media communications plans with fact sheets and talking points for a variety of National Weather Service program areas with a specific focus on weather and hazards that the state of Hawaii faces. And this project was done in support of the operations of the Honolulu Forecast Office and the Central Pacific Hurricane Center. So with this overall goal in mind, the actionable goal for my project was to create simple yet thorough media guides for a variety of weather events so that when these events happen, there's a ready-to-go resource for interviews with media partners. And my hope was that these guides could be designed to be used by both media partners, such as reporters, and also NWS staff who may be giving these interviews. They would help media personnel be more informed ahead of interviews and act as a resource of fast facts for NWS staff who may be giving these interviews. So my personal interest in this project stems from the fact that in the field of meteorology, there are many different paths you can take. At school, I'm involved in research, and I've had the opportunity to see what that path looks like. And so for the summer, I wanted to try something different, and this project gave me the chance to explore the communication side of meteorology. And I believe an essential part of meteorology is the accurate communication of scientific knowledge to the public in the face of extreme weather events so that everyone involved can be more prepared. And this project in particular had an element of collaboration between the National Weather Service and local media that was a great opportunity to see a real life scenario of the importance of communication. And working at a weather forecast office also provided me with the opportunity to see the operational side of meteorology. So to understand the scope of the media guides created for this project, here's some quick background information on weather in Hawaii. So for most of the year, weather in Hawaii is dominated by trade winds. And these trade winds are the result of the North Pacific High, which is a semi-permanent subtropical area of high pressure in the North Pacific Ocean. So the location of this high, semi-permanent high to the northeast of the Hawaiian Islands means that the air circulation passes the islands as northeast trade winds, since airflow around areas of high pressure is clockwise. So these trade winds are a persistent feature of Hawaii's climate due to the permanence of the high pressure system. So in the summer, the high reaches its northernmost position, bringing the area of the trade wind zone directly across the state of Hawaii from May to September. And then from October to April, these trade winds kind of lighten up as the high moves a little bit further south, and it's just not as strong. So Hawaii also does have a wet and a dry season. Hawaii's wet season runs from about October to April, and during this time we see cold fronts and Kona storms bring heavy rainfall to the islands. So as I just mentioned, Kona storms bring wet and stormy weather to Hawaii. These are subtropical cyclones that form from about October to April in the North Central Pacific. They kind of form from like kinks in the jet stream that kind of pinch off and then move towards the islands. The Hawaiian word Kona means leeward, which describes the direction of winds that result from these storms. The winds that are associated with these storms move in from the southwest or Kona side because of the counterclockwise rotation of low pressure systems. So these winds replace the usually persistent easterly trade winds, bringing wet weather to typically drier leeward sides of the islands. And then also, due to Hawaii's location in the tropics, hurricanes are a threat that the state must be prepared for. And Hawaii is in the Central Pacific Basin, which runs from 140 west to about the international date line. And formation of tropical cyclones within this basin doesn't happen too often, with Tropical Storm Emma being the last to form in this basin. However, hurricanes do often cross from the Eastern Pacific over into the Central Pacific, as we saw as recently as this past week with Tropical Storm Calvin. So these storms do happen, and the state does need to be prepared for them. And one important relationship to note is that El Nino typically causes an increase in tropical activity in the Pacific due to weaker upper level winds, which reduce wind shear, which pair apart hurricanes. So without this, they are more frequent. Next, there are a few more weather events that impact the state of Hawaii, and the next being winter weather. Despite Hawaii's tropical location, winter weather does occur on these islands since there are high elevation peaks. Winter weather in Hawaii is generally confined to high elevations, affecting really only Maui and Hawaii counties. Mauna Kea and Mauna Loa both have elevation over 13,000 feet, and snow does happen there about every year. Meanwhile, Haleakala on Maui reaches just over 10,000 feet, and snow is a little bit more rare there, occurring only every 2 to 3 years. And while no permanent residents live in these impacted areas, it's still important to note that winter weather can occur even in a tropical location. And then lastly, Hawaii faces air quality threats that are tied to volcanoes. The first one being what's called VOG. So VOG is a combination of the words volcanic and smog, and this hazy air pollution is caused by volcanic emissions from Kilauea volcano. So VOG is composed primarily of water vapor, carbon dioxide, and sulfur dioxide. Of those pollutants, sulfur dioxide does pose the most hazard to human health. So the state has developed a SO2 color code advisory system to warn residents on Hawaii Island of volcanic SO2 pollution. And then lastly, just want to note that during and after volcanic eruptions, volcanic ash can also pose a threat to air quality that can cause air quality alerts to be put out. So as you can tell, the most important piece of this project was understanding what kind of events impact Hawaii and its residents. I spent a lot of my time researching various weather events, and during my research, I used various NOAA and NWS websites. I used Weather Forecast Office websites. I used NOAA media guides. I used local news articles. And I spent a lot of my time also foreshadowing various forecasters at the Honolulu Forecast Office, and their insight into common weather patterns and their knowledge of the islands was key in helping me decide what information to include in my media guides. And then lastly, I did just want to note that another important resource I used was the Bishop Museum website. The Bishop Museum is Hawaii's designated state museum of natural and cultural history, and I use the Bishop Museum as my source for knowledge on Hawaiian names of various weather phenomena, including Kona storms and also Lahaina noons. So to gain further insight on specific focus areas, I attended various meetings with experts in these fields. To learn more about air quality threats specific to Hawaii, I listened in on an interagency call about VOG with staff from USGS, NOAA, NASA, the National Park Service, CDC, and Hawaii's Department of Health. And then at the end of June, I also had the chance to attend a few meetings with Crane Johnson, the hydrologist in charge from the Alaska River Forecast Office, as he visited HFO. And in those meetings, I learned a lot about hydrology, flooding, and also the various tools that NOAA uses to predict flooding events and how that risk is communicated to the community. Lastly, a key part of this project was understanding what happens when weather becomes news. And to gain this understanding, I met with some local media partners here in Honolulu. I had the chance to observe some reporters in action as well. Local TV stations often work in collaboration with the National Weather Service in the face of big weather events to deliver key messages across the islands. And I watched as reporters came to the NWS office, where they recorded interviews ranging from topics to ENSO, as recently as 2 days ago, Tropical Storm Calvin. And I also had the chance to visit KHON downtown and see the newsroom firsthand. So at the end of my project, I have created 11 media guides covering a wide range of weather events that impact Hawaii. The topics that are covered by these guides are seen listed here, and there are a few things I did just want to point out. You may notice how impacts of climate change is one of the topics I covered with my guides. While climate change is not one specific event, it's a hot topic that often gets brought up in interviews, and therefore I wanted to include it in my list of topics covered because I think a greater understanding of climate change is beneficial for everyone involved. And while this guide includes some basic information about climate change, it also offers some suggestions on how to navigate questions from the media about climate change and climate science, especially for scientists whose official work is weather and water forecasting, not long-term climate science. And the other thing I just want to note was the topics that were covered under public and severe weather were lightning, hail, high noons, waterspouts, trade winds, and tornadoes. This is my kind of catch-all topic where I wanted to cover events that aren't very common or don't pose any threat to public safety, but still may be asked about during an interview. So this was the layout for my media guides. The first section was weather talking points, where I covered what topic each guide was about and any background information on events that may not be as well understood by the general public. The next section covered what kind of products are issued in relation to this kind of event and the conditions that need to be met for different products to be issued. So like what constitutes a watch, what constitutes a warning, an advisory, an alert. Next, each guide covered how the event was related to Hawaii and the impacts that residents may feel or have historically felt in the past. And lastly, I covered some frequently asked questions that often get brought up in media interviews relating to each topic. I did just want to note that not all the guides followed this layout to a T, but it was the general structure that I used. And the length of my guides range from 1 page to 4 pages, with the hurricane guide reaching 7 pages, as it included a list of tropical storms that have formed in the Central Pacific Basin over the last 25 years. So this project was in support of NOAA's Weather Ready Nation initiative. Weather Ready Nation's goal is to help our nation become more resilient to increasingly extreme weather, water, and climate events. This is done through partnerships with community leaders, such as emergency managers, local media, and businesses. And these leaders are often people that inspire others to be better informed and prepared, helping to minimize or even avoid the impacts of natural disasters. And a Weather Ready Nation takes well-informed communities, businesses, and individuals that are ready, responsive, and resilient to extreme events. NOAA is hoping to create this Weather Ready Nation through a network of ambassadors, which is made up of local leaders across the country contributing in the ways they can. And this is connected to my project through the involvement of media partners. Media partners, and especially reporters, are a crucial piece in getting information about extreme events out there to the public. Their on-air advocacy for preparedness in the face of extreme events is crucial to creating a better informed and better prepared general public. And the hope is that these media guides that I have created will aid in the goal of creating this Weather Ready Nation. And these guides give media partners a better understanding of events that impact their communities. Because ultimately, when media is better informed, they can better inform the community and thus create a Weather Ready Nation. So in summary, these guides are really important because information that those in the field may consider to be obvious may be unknown to those outside of the field. And my hope is that these guides will provide key background information to better inform media partners. And again, to kind of beat a dead horse at this point, when the media is better informed, they can better inform the public. And the greater the understanding of weather events and hazards that the media has, the more informed and prepared the general public will be, which isn't something that can be overstated. And lastly, I did just want to note that these are not meant to be authoritative sources of information. They were created with the purpose of being internal resources to be used for media interviews. So ultimately, the next step for this project is its implementation. So my hope is that these informational guides will be used for media interviews at the Honolulu Forecast Office, both by media partners and NWS staff. And another note is that some elements of these guides are 2023 specific. So if they are to be used throughout the future, they'll just need to be updated annually. And personally, my next steps, this year I'll be finishing my bachelor's at the University of Miami, studying meteorology and Spanish with a minor in mathematics. And I'm interested in possibly attending graduate school with focus in GIS, emergency management, or as this summer showed me, I do have an interest in communication. So I just wanted to thank John Bravender, my mentor, who is the warning coordination meteorologist at the Honolulu Forecast Office, the entire staff who let me shadow them, and the Hollings Undergraduate Scholarship Program for being the funding sponsor. So thank you.
Great job. Thank you so much. Do we have any questions? We have time for one question. All right, in front here. Microphone on its way. Working up.
So you mentioned that your media write-ups were not there for internal use, not for public outface. Is there a resource like that that does exist? I could see this being used in one of my future potential classrooms.
When I was conducting my research to create these media guides, I found a variety of resources that NOAA has already produced that are meant for general distribution. They are a little bit more general. So the ones that I made are kind of more specific to answering those questions for media. So they really weren't designed to be like educational. But there's definitely things out there, and this project could be adapted in the future to kind of be more like the purpose being like general consumption. So yeah, thanks.
All right, thank you so much. That was wonderful. It's always good to see. Yeah. It's always great to see people interested in communicating the science that we do, because as scientists, we have a tendency to get wrapped up in the facts and details, and communicating them to the public is not the easiest for us to do. So it's great to have people creating things like guides for us that we can use to help communicate the public, because it really is all about getting information out to everybody. All right, next up, Haley. So Haley is going to talk about Pacific GIS Research Cruise Operations Dashboard.
Okay. Good afternoon, everyone. My name is Haley Chong-Ki, and I am a current PISO/Hokuala intern with Kimberly Lowe as my mentor. I am currently studying environmental science with the School of Ocean, Earth Science, and Technology, also known as SOEST, at the University of Hawaii at Manoa. My project was to create a user-friendly online GIS dashboard. For those wondering, a dashboard is a way to overlay geographic data from different sources on a map with visual tools like widgets to make it easier to understand. And these are the steps I went through in order to create the dashboard, and that I will be showing you for today. I started with no GIS training. So during the first couple of weeks, I spent my time learning about the different terms and techniques of what GIS is. I also took a one-day workshop on how to create and design an aesthetically pleasing dashboard. Just to keep in mind, anyone can do this training as it is already paid for by NOAA, as long as you have a NOAA.gov email or ArcPro downloaded. When completing the layers for our base map, I built on others' works, such as looking for new layers to replace old ones. Some of the layers we had to add were recent fisheries independent survey system, also known as FINS. I had to add different science centers, such as oh, well, I don't know why it's not working right now, but I had to add different science centers such as Northwest, Alaska, Pacific Islands, Southeast, and Southwest science centers. And then there was supposed to be a transition to where it shows the different locations of all those science centers. So after we collected our layers, I was then given access to our base map. Some of the things we had to do was reconfigure the pop-up names and add info data layout and add symbols and group the layers based on the categories for better navigation of finding the layers. So I was supposed to show a picture of our dashboard, however, it's not showing up as well. But after forming our base map, we then made a layout of the dashboard of what elements we wanted, such as the number of active cyclones in the area, a certain display of vessel data, and a gauge of active cyclone strength. And I was going to show our sidebar, which explained how to use the dashboard. And then it was also going to show that you can expand the elements on the dashboard to make them bigger. And then it was going to show you a picture of a certain buoys layer where it showed Oahu and it showed different locations of different types of buoys, like submerged and fins data. And then I was going to expand back out to show you the different ships that our dashboard is looking for or researching. And those are some of the examples in case people were interested in seeing what it actually looks like. Lastly, after completing the dashboard, we had a meeting with people who would use the dashboard or who helped us with it along the way. So we asked them for their opinions and feedbacks and improvements for the dashboard. And then in conclusion, our research cruise operations dashboard is not only a good way for scientists and others to visualize data, but it is also a good educational purpose to teach others about NOAA research vessels. So thank you for your time. And if you would like to see our dashboard in person, you can use the QR code here, or if you have any questions or comments after the presentation, you can email me or Kimberly. But does anyone currently have any questions or comments at the moment?
Is it possible to bring up the dashboard just online there so we can have a look?
No.
Whose computer? Who are you logged in? It's showing at the bottom. It's not. It's projecting. Yeah. Maybe if I could say we do have the dashboard, it's operational. Kate is actually going to be here for a couple of weeks in the library. There's a workstation there, and you could drop by, in the late afternoon maybe, and just actually get a demo. We'll probably do another demo of the dashboard itself. Yeah, unfortunately, some of the slides got rearranged, and it seems like they didn't show up the way they were showing up.
Yeah. Well, you still did a great job.
Thanks.
Yeah, this is what happens.
Science and presenting and every day. I actually do have a question though. Is this updated in real time? Like I noticed you had the Rainier out there. So if we look today, would we be able to see
where the Rainier is.
You know what, I
Yeah.
can pull it up on my laptop and maybe share it later on.
Yeah. Yeah. That's great. Thank you so much.
Thank you.
I was. Yeah.
Yeah,
I can. Yep.
Yeah, we have some time. I was just going to say that,
Oh, yeah.
Haley has been an amazing intern. She's learned, as she mentioned, she had no GIS experience when she started, and she just learned a whole lot of skills in a very short amount of time. Her ability to click and change everything, which most people in her generation have, and I don't, is amazing. And so she quickly got up on the dashboard and was configuring and making things look beautiful. So I really hope you will get a chance to see the dashboard in operation. It's really phenomenal. And we were watching Hurricane Calvin coming in and watching it get downgraded and seeing the live weather and where the ships are. So, you know, it's to let us know where the ships are and what are the conditions where they are and what's the depth and where are the reefs and where did we survey before. It's a really valuable application, and she put a lot of work into it, did a really good job. Appreciate that.
All right. Next up, we have a virtual presenter, Hugo. Hugo is going to talk about flash flood mechanisms in Guam.
Alrighty, I hope you guys can hear me okay. I am Hugo Richardson. I am a rising senior at Dartmouth College, currently doing a Pathways internship with the WFO at Guam. And before I start, I would just like to acknowledge my mentor, Brandon, as well as Dr. Habana at the University of Guam, and the entire WFO team here in Guam. So to start, I thought I would just give some general context. Here's Guam. It is the southernmost and largest island in the Marianas. Zooming in a little bit, we can see that all of the streams are concentrated on the southern half of the island. The northern half is like a big limestone plateau. It's very porous and permeable, so it soaks up any water like a sponge, whereas the southern half is volcanic in composition, meaning it can have surface runoff, and the streams are very steep, making them susceptible to flash flooding. I'm just going to go through a few examples of floods in Guam to give you guys an idea of the damage it can cause. These photos are a result of damage during Typhoon Pongsonhua. This is from Typhoon Mawar in 2023, back in May. The Pago River overflowed its banks and undercut the structure, causing some damage. We actually don't have images from that flood in May, but I was able to recreate its extent using ArcGIS. More recently, even in July, there was a stream that overflowed its banks in Marizo just last week, and a similar flood happened in Agate back in August of 2020. That same August 2020 event put this guy's house 5 feet underwater and actually took a car out to sea. And on top of all of that really obvious infrastructure damage, there has been some very high-profile drowning deaths associated with flash floods back in 2014 and 2018. And if you talk to anyone on Guam, they will tell you numerous anecdotal stories about flooded roads and bridges and fields. Flooding is a very pervasive issue. And despite this very big impact of flooding, we actually don't understand flood hydrology very well. Dr. Mark Lander, a meteorologist at the University of Guam, in response to those drowning deaths, said some very basic questions remain unanswered. For example, what is the threshold of hourly rainfall at which a dangerous flash flood is sent down the Longfit or Tarzan rivers? And we actually don't understand this for any of the streams on Guam. So that brings us to our research question. which is how much rainfall is needed to produce high stream flow in Guam streams. Lander was interested in the hourly time scale, but I was curious over what time scales does rainfall matter. Can we have a lot of rain in 15 minutes and that produces a flood? Or do we need it for an hour or even longer? Does it depend on the antecedent conditions over the past few days? And then does this result vary spatially? The second to do with verifying the WFO's warnings, watches, and advisories to see if they've done a good job predicting these floods, but I haven't gotten to this yet. I still have a month left of my internship. My hypothesis was that if over an inch of rain accumulates within an hour, streams in southern Guam will experience a high flow. Here's a brief overview of my methods. I will go through each of these steps in detail, but the thing to note is that this was all completed using R and ArcGIS Pro. The first step in this was understanding what data is available to us. Here is a map of my study area. There are currently 6 USGS stream gauges on the island, which report the discharge of a stream every 15 minutes via satellite. And there are 8 rain gauges, but only 4 of them were close enough to the streams to be useful to us. Also shown in this map are the watersheds, which is basically any water, any rain that falls in those pink blobs will make its way and contribute to the stream flow at each of the stream gauges. If it wasn't clear from the last graphic, 2 of the drainage areas, the watersheds are much larger, being that of the Pago River and the Ugam River. This has some implications hydrologically, so I consider them separately in most of this study. And another thing to note is that generally speaking, in these hydroclimatological studies, we like to see about 80% or greater for record completeness, and we are sitting well above that at all of our gauging stations, which is pretty good. And I start data collection in October of 2007 because October 27, 2007 is when all of the gauges were hooked up to the satellites to report instantaneous data. My next step was to link rainfall and streamflow records. From the 2007 to 2023 period, there was over 5 million raw data observations, and the satellites all report at different times, so I had to do basically some data massaging to link the records and make it so that for each timestamp, we had both rain and a stream flow observation. Once we've compiled our data, I had to go through and identify high flow events or the floods. What stream flow conditions do we actually care about that could impact people? Here is a graphic just providing an example of how I did that. On the top, we have hourly precipitation accumulation versus time. And on the bottom, we have the river discharge in cubic feet per second. This is just one example from the La Safua River in August of 2020. So the first thing I did was use a peaks algorithm to identify any peak flow above a threshold. And all of these peaks are shown in red. And then I went and took the top 10% of these peaks over the entire record. Those are shown in yellow. And basically, those are the floods that we care about. I did this for each of the stream gauges over the entire record. And just as a sanity check, I showed you guys this flood earlier from August 27th, 2020 on the Umatic River. The La Safua River is just a mile away, so we would expect a flood on that as well. And as we can see, yes, there is a flood, and this process I'm doing seems to do a pretty good job at picking up on many of the major floods. Just to give you guys a better idea of how these events are temporally distributed, these rose diagrams show when in the year the floods are happening, and they are all happening, or most of them are happening during Guam's wet season, which happens from July through December. This is for a few reasons, ranging from general increased atmospheric moisture, a decrease in wind strength, meaning that showers can park over the island for a little bit longer, And also tropical cyclones are much more common during this time of year. So now that we've assembled our data and identified the floods, it's time to actually answer our research question. What precipitation conditions contribute to flooding? The way I did this was by first using an extreme gradient boosting machine learning model. And that sounds like a lot, but basically what it does is I give it a bunch of variables, which in this case is antecedent precipitation conditions. I give it 15-minute precipitation totals up through 30-day precipitation totals. And it will make a model to predict whether or not there's a flood. After it makes the model, it tells me which of the variables was the most important in predicting flood conditions. So for the Emong River, this is the example I have on the screen. 1 hour precipitation accumulation was the best predictor of flood conditions. It turns out that all 4 of these smaller watersheds follow this exact same behavior, whereas the 2 larger watersheds, the Pago River and the Ugum River, are more dependent on 2-hour precipitation. As well as this extreme gradient boosting machine learning model, I also ran this analysis using a normal multiple regression model, and it replicated the exact same results. Just to give you guys an idea of how powerful this tool can be in helping us understand when floods occur, from over 140,000 observations of stream flow, the model was correctly able to identify the one impactful flood. The model is trained to tease out really rare events. They've used it in hadron colliders before, and I think it's pretty cool that we are able to use machine learning to understand flood hydrology. Now that we have gained an understanding of what timescales matter, it's time to actually put a number to that. So here we have a graph of antecedent precipitation over various timescales in inches versus frequency. This is basically showing for each flood, what is precipitation doing? And we can see that for the small watersheds, which are dependent on 1 hour precipitation, it takes about 1.12 inches within that hour to generate a high flow. For the larger watersheds, it takes about 1.34 inches over 2 hours to generate a high flow. And something I would like to point out is that, these smaller time scales, 15, 30, 45 minutes, can have a lot going on. You can have no precipitation in the last 15 minutes, all the way up through 0.6 inches. And it doesn't really have a great central tendency, which makes sense. It's not a great predictor of flood conditions. And then looking at the other end of the spectrum, the 3 days through 30 days, we can see, yes, there has been some antecedent precipitation, about 5 inches in the last 3 days, 7 in the last week. This is basically confirmation that these floods are happening in the wet season while soils are saturated, which is a sort of necessary condition for the genesis of floods. Take-home messages are that small streams are most responsive to 1 hour precipitation accumulation. High flow events are associated with about 1.12 inches of rainfall within that hour. And large streams have more lag time, making them more responsive to 2 hour precipitation accumulation. High flow events are associated with 1.34 inches of rainfall over those 2 hours. And something to note is that we can't necessarily use this to guarantee that a flood will happen. It doesn't mean that every time we get an inch of rain in an hour, there will be a flood. This is just looking at past trends. And with that, I will take any questions.
Great, thank you for that presentation, Hugo. Do we have any questions in the audience here? Anybody online? Any comments there?
No comments online.
Great, thank you. Somebody online? No?
So we do have Hugo's mentor online, and he's going to say a few words.
Hey, good morning, everybody. I was just able to log in time. So I just want to say this is a really interesting project that Hugo has really gone into a lot of dimensions with this, taking his hydrological background and really applying those skill sets to really take a deep dive into Guam's hydrology. And so that's been one of the long-standing questions dealing with the rainfall, the stream flow response, and really seeing what does it mean and how do we tie in the data that we have with the stream gauges and then relate that to impactful flooding. And so that we can then in the future kind of take this information and categorize and really assess what kind of rainfall events lead to that impactful conditions. And so I just want to say a good job to Hugo for just his diligence in keeping up with this. And so I'll turn it over back to you all.
All right, great. Thank you so much. And next up, we have Michael. Michael is a Hollings scholar who is going to maybe walk us through an interactive virtual tour of this beautiful facility, the IRC, here on Ford Island.
Well, good afternoon. My name is Michael Lee and this summer I had the wonderful opportunity to work on building an interactive virtual tour here of the Noye Regional Center on Ford Island, Oahu. Under the direction of Leon, Jeswin, and Mike Pye, my mentors, I was able to craft a virtual tour to fully describe and visualize this space in an online setting. The ultimate goals that we had in mind behind our whole project, right, was first that we had to convert a face-to-face tour to a virtual setting. So we currently are giving face-to-face tours here at the IRC. That is something that currently is an outreach product that we have. However, it's oftentimes difficult. As we all know, we work here at the IRC or additional NOAA locations. And as such, it becomes difficult to get outside visitors onto here, especially on Ford Island, where we're behind the military access badge. And so it becomes a lot more difficult for outside visitors to get in here, making it difficult for face-to-face tours to occur. So we want to, convert this face-to-face tour to an online setting. We also want to provide educational and outreach tools to teachers and Hawaii students, along with the general public, so that they can see the work that NOAA does here in Hawaii and the importance behind all the work that we do. Personally, I'm pulling on the background of an education and science background. I'm currently studying my Earth Science Education BSE from Millersville University in Lancaster, Pennsylvania, as well as my media arts background, which I get from working as a professional wedding photographer back home. There it is. My mentors, Leon Jeswin, is our education and outreach specialist here at the IRC, and Mike Pye, an animator. Leon really gave me the opportunities to work on how to give a face-to-face tour. I had the opportunity to step in and give a number of tours here at the IRC and see how that works in a face-to-face modality. As well, Mike Pye helped with a lot of those creative decisions. I don't have a huge graphic arts design background, and so getting his feedback was critical to making the end product just look pretty, right? And like I said before, we want to step away from a bit of the face-to-face. Not only is it difficult to get on base, right? You have to have all those extra passes in order to get on here, but it's also a lot of personnel and timing issues. We face a lot of issues where we don't have many individuals who are ready to give a tour, or the students and schools are working under strict constructs that only allow us to provide tours for certain amounts of time. And so we're not able to fully show off the entire depth that the IRC covers. And so presenting a tour in a non-face-to-face modality and instead in a virtual space was critical to kind of solving those issues. But what does that even look like? What does a virtual tour look like? So a virtual tour in a simple place is taking a 360 panoramic photo to be able to fully look around and see the entire virtual space. Unfortunately, I do not have any videos today to show you, but later today you'll get an opportunity to scan a QR code, yes, and you'll be able to look around into the IRC itself. As you can see from my photo behind me, you're going to notice this overlay that I've placed at the top to help with ease of access and moving around. as well as having these blue buttons that have now changed in a couple of different ways. But they still exist as a way to move between different panoramic photos and fully walking through this space as a whole. If you're imagining a Google Street View cam system, right, that's a very similar setup to what I've worked with here in the IRC. And anything that you will see in the tour has been fully crafted by myself. The equipment that I used to build this whole setup was to start this 6-lens Insta360 Pro 2 camera. This is a 6-lens panoramic photo camera that shoots with all 6 lenses twice. The resulting effect is a panoramic photo in stereo mode, which provides a 3-dimensional effect if you're looking at it in a virtual reality headset. Along with the Insta360, after I shoot my photos on that camera, you'll be importing them into Insta360 Stitcher, which is a stitching software that takes all those 6 different lenses and puts them into one viewable image. Once I have that, I take all of that work, which takes about 3 hours between shooting and stitching together, and throwing it into 3D Vista, which is the process of stitching all of those different images together so that you have the buttons to move in between everything and the idea of how to look around in the space in the virtual world. So a lot of my work goes into 3D Vista in that post-production steps. Afterwards, I upload our server to a temporary hosting service, which allows me to send out links so that you guys can gather feedback and I can see how it actually works on different devices. The end product can be used on a computer, a mobile device, as well as the virtual reality headset in order to fully realize and see the space that we work in here at the IRC. Now, along with just learning that equipment, that was my week 1. To learn all of that equipment, Leon had no idea how to use it. It was terrible. It was miserable. So after I got to do all that, right, now I have to build my interview list, right? I'm connecting with NOAA scientists all across the IRC. I'm learning what spaces I need to get into. I need to build this massive interview list to fully realize this huge space with all these different people who are coming together to do it. So once I've connected and built that interview list, I'm reaching out. I'm talking to actually most of you who are sitting in this room. I think I've talked to a lot of you, and figuring out ways to get into the different spaces around the IRC. Once I do that, I'm holding interviews with you in your workspace. You're letting me into your busy schedule to come in with this big bulky camera and say, yeah, you need to let this little intern come around and show you what to do so I can show it off in a virtual space. It's chaos, but it's a lot of fun. So now that I have those interviews in places, you know, I'm going through my camera and I'm taking those photos. I'm playing this game of hide and seek where I set my camera up to play and then I run away from it so that it can take its photo. And then once I have all those photos taken, I've fully shot the IRC. It's time to go through that editing process. Now, this takes me the bulk of my time, right? So I'm taking those photos off of the 360. I'm setting up my computer to run with Stitcher. I'm moving them into the post-production. I'm putting all my buttons together. And then at the end, I have to sit for 3 hours as my computer exports the final product. Miserable. And then on top of that, I have to spend 18 hours uploading it to the Amazon server, and it's just miserable every step of the way. But I had a good time, I promise, I swear. Leon really treats me well, I promise. Anyway, now it's not just done once I have everything exported and uploaded. The final step is the gathering feedback step. Now, this means that scientists aren't always happy with the work I do, which is fine, right? Sometimes I get it wrong. But that means I need to make sure I improve on it and go through this reiterative process where I get your feedback on how to improve and better show off the work that you do. Now, this oftentimes means that I go through this whole process again and again and again. However, the end product speaks for itself, and I think I'm very proud of the work that I've done in order to make sure that I'm showing off this space in its full entirety. But now, for the big reveal, this QR code behind me, if you were to scan this, will pull you to, I believe actually our previous version of our draft tour. The end result is a fully realized draft tour of the IRC. It shows off all the spaces that we have in here that we deemed of importance, our different lab spaces, including our vet lab, our acoustics lab, our necropsy lab, our fish and turtle necropsy lab, our dry labs upstairs, along with our maker lab. It shows off our small boats and boatyard spaces, our B-130 warehouse, our dive locker, our small boats, our Oscar Rainier and our Oscar Seti and Rainier ships, along with our educational center, which includes the Science on a Sphere, NOAA's career banner, or Leon's career banner space, the NOAA library, along with the lobby space, along with our tsunami centers, which includes the International Tsunami Informational Center upstairs, along with the Pacific Tsunami Warning Center upstairs. All of those spaces shot by myself, talking to those scientists, going through that whole process to make sure I can put all of those into it. And final product is now online and accessible. Scanning this QR code will bring you actually to the previous iteration. Leon sent me an email 10 minutes ago that his computer had finally finished uploading our next iteration of the process. So I have that ready to go as well. And the final product looks pretty good, I think, right? It's perfect for live guided tours, outreach events, ways to show off the IRC on a little more of a, when you're harder to get into the space, you can still view it online. However, it's not perfect yet. It still has some room to grow. But it is also perfect for orientation for new hires, right? If you have a new staff member coming in and you want to show them off a little bit about the IRC, but they, are struggling to get on base, you want to do it before they even get here, send this ahead of time. It's perfect for them. They can get to see and visualize the space before ever physically stepping foot into the Inouye Regional Center. But that means we have plenty of room to grow. The future of the tour would look like pushing us away from that live guided section and more to a self-guided tour. That's adding in a lot of context, which requires me talking to a whole new slew of scientists and getting them to add their little notes about everything that you're seeing in the tour and how that we can write it down and have it ready to go for the context markers that would come through with an updated tour. It also would be perfect to find a way to better fit it in with a classroom environment, including lesson plans and activities for teachers and students to use in a classroom on how they can use the IRC tour to get an understanding of NOAA's work and better understand some of the science that we do here in Hawaii. My personal future is going to be a little bit chaotic. I go back to Silver Springs on next Friday to present this work at the Hawaiian Symposium. I also will be presenting my work at Millersville University in our Made in Millersville Symposium. I also will be presenting my work hopefully, at the Ocean Sciences meeting in 2024 down in New Orleans. And after all of that, I also have to start my student teaching this fall, which I'll be doing for the working with a classroom of 9th and 12th graders in Conestoga Valley High School back in Lancaster, Pennsylvania for a full year. I'd like to give a special thanks to Leon Jeswin, my mentor, for all the ability that he gave me and all the help he did connecting me with everyone, along with Laura Cummings, Michael Pye, and everyone else who I was able to interview to be able to get to build this final product. Without you, I wouldn't be able to do anything that I have done, and I wouldn't be able to realize the virtual tour that we have today. I'd also like to give a very special thank you to the Noah Ernest F. Hollings Undergraduate Scholarship Program for my funding sponsor and sending me out here to spend a summer in Hawaii. Thank you.
Awesome. Very, very cool. Do we have any questions? Yes. Hold on 1 second. Microphone's coming.
I'm not that fast.
Oh, thank you for your very dynamic presentation. And I was wondering if you did get into the laboratory necropsy areas and stuff. I think there are people that will be so happy to have this virtual tour available when somebody wants to see those spaces.
Yep. So those spaces are fully realized. We show off the fish necropsy lab along with the full steps with included in a fish necropsy. It is a little bit gory, but hey, it's realistic. And we also have our turtle necropsy lab showed off. Now this is a little more cultural, sensitive. We only have our turtle models on display. I don't go through a full turtle necropsy with all that smell and everything, but we show off those spaces in their entirety and their use cases and how we use them here at the IRC. Yep.
What is the narration or the over talking on the tour like? Is it just you talking? Is it the scientist talking, or is it kind of a mix of both?
So right now there isn't a audio aspect to the tour by itself. There are some videos that I've included, especially in the Tsunami Warning Center, where we have some interviews of duty scientists talking through how they use their space, what they do in it, some of the background behind why it's important, and some of those aspects. The end goal would be including a lot more video that does show off scientists in their space talking about the work that they do. I don't want it to be me-centric. I want it to be you-centric because it's your place of work. And you're the ones using it, right? And it's boring to listen to me talk about everything where I could just have you guys talk about your individual spaces. So the end goal would be you're talking about your individual space at the Weather Service, and then someone else might talk about their uses in the dive locker or the lab spaces. Yep.
So did you teach Leon how to use the camera?
Yeah, that's my next week issue. Yeah.
I just want to make a comment as someone that works in outreach and education. This is so useful. So we really appreciate this. As you know, it's very difficult to get people out here. And so to have this as a resource is really valuable for all of us, really. So thank you.
Thank you.
And we get
Just one more comment about this. I'm really excited about this as well. My nieces live in North Carolina, and I cannot wait to show them the facility, and especially the tours of the NOAA ships, getting on there. That's amazing. I tell people about the research cruises that I do, and it's going to be really awesome to be able to show them the actual ships. So great work. And Leon, you want to come up and say a few words?
So to Ray's question about whether I'll be able to use the camera, yes. So Michael's actually I've asked him to write standard operating procedures for everything. So not only will I be able to use it, but hopefully everyone will use it for outreach. It's an outreach tool that anyone can use in the building. So that's first off. So if you want to shoot your space, there's a possibility of expanding it. Back to Michael. I mean, he's been such a joy to work with this summer. I mean, he basically jumped into anything that I said, oh, why don't you try this? Do you want to give a school tour for something? Sure, I'll try it. So I think there's that enthusiasm and that infectious, just to wanting to jump into any situation has been really contagious. It's given me a great opportunity to meet a lot of people that I haven't gotten to their spaces. So I think in having this project, it's built a lot of relationships within the IRC for myself. But I think in general, Michael's done an incredible job just from the perspective of not only handling the technical side of things, but also working with scientists, staging them. Okay, where do I stand? Oh, how long? Oh, they're speaking for 20 minutes. Okay, can we cut it down to 2 minutes or 3 minutes? So managing all that is a difficult process, and he did an incredible job in doing it. So kudos to our experience. I'm very grateful for the summer experience. So thank you. Yeah.
All right, our next speaker is Hannah, and she is going to talk about finding trends within different types of meteorological data during Typhoon Mawar.
Okay, hi, I'm Hannah. I'm one of the Pathways interns for the National Weather Service from this summer, and today I'm going to be talking about finding trends within different types of meteorological data during Typhoon Mawar. So just to give you a little bit of background on the storm before I go into it, is that so far this year, it's been one of the most intense tropical cyclones. And then it's also been one of the strongest in the Northern Hemisphere during the month of May on record so far. And it's also one of the top 10 strongest since 2005. It had sustained winds of up to 185 miles an hour, and it had a minimum pressure of 897 millibars. And as Mawar passed by Guam, it gave the central and northern parts of the island over 2 feet of rain. So this is a view from the Himawari geostationary satellite of Mawar approaching Guam, which is outlined. And then this is showing Mawar's track as it approaches Guam. So the path is the line, and then the colors show the intensity of the storm. So it starts at the bottom in the purple as a tropical disturbance, and then it intensifies all the way into a super typhoon, which is shown in pink. And as Mawar passed by Guam, it briefly made landfall at Andersen Air Force Base, which is on the northernmost tip of the island. And it did this at 9 p.m. on May 24th. And when it made landfall, it was the equivalent of a Category 4 Atlantic hurricane. And then after it progressed past Guam, it re-strengthened back into the equivalent of a Category 5 Atlantic hurricane. So after Mawar passed by Guam, it left 150,000 Guam residents without power or utilities. It uprooted trees, it ripped the roofs off of houses, but luckily there were no fatalities and recovery efforts are ongoing. And these are some images to show you the destruction that it caused. So what I did is the first two types of data that I looked at are pressure and wave height. So the pressure sensors that I used are shown on this map in the blue markers, and then the wave height buoy that I looked at is in green. So the devices that I used to get my pressure data are from 9 smartphones that are distributed throughout Guam. So all of these smartphones had the app RedFox on them, and the primary purpose of this app was to record infrasound, which I'll talk more about later in my presentation. But they were also able to record the pressure changes. So this is showing the changes in wave height as well as the changes in pressure. So the wave height is the thinner dark blue line, and then the other thicker lines are the 9 different pressure sensors. So just from looking at this plot, we can see that on the left-hand side, the wave height seems to be very clearly elevated and then drops back down. And then afterwards, we see the drop in the pressure. But just because this plot is shown on such a short time scale, it's not clear that this is not just the usual ups and downs. So to make sure of it, I wanted to plot the wave height on a longer time scale. So we can see that during the typhoon, the wave heights really jumped up past what's their normal, reaching a maximum of about 6 meters. So then going back to this plot, what's interesting to note is that the wave height reaches its maximum a little bit before the pressure reaches its minimum. So one potential explanation of this could be that the wave height maximum was reached as the eye wall of the storm passed, and then the pressure reached its minimum as the eye of the storm passed. Another potential explanation, and something else to note, is that the wave height buoy sensor was not located in the exact same location as the 9 different pressure sensors were spread out. So that can also lead to some discrepancies. So I didn't have access to one sensor that could record all these types of data from the same exact location, but I did have access to a third wind speed sensor, which is shown in red. And it's also important to note that these are in 3 different locations. And then instead of using the 9 pressure sensors, I decided to pick just one that was in the closest proximity to the wind speed and the wave height. So here I have shown all 3 types of data at the same time. So the dark blue is still the wave height, and then the light green is the pressure again, and then the lighter blue is the wind speed. So this is also a slightly different time scale than the previous plot. So we can clearly see the rise and then the fall of the wave height, and then the same drop in the pressure. And then we also can see that the wind speed reaches its maximum after the maximum of the wave height and then the minimum of the pressure. So they did not occur simultaneously. So one of the reasons for this could potentially be because they changed as Mawar moved and its path, and it moved in a different direction from these sensors. So we can quickly look at this. The time zones are wrong, but we can see that Mawar is moving around Guam. So because the 3 different sensors have different relationships into where the storm would be passing, that could be one of the reasons why. Yeah. And then here is just the path again, so we can see. And one other thing to consider is that as Mawar moves in relationship to the sensor, we would expect the wind direction to change. So the wind direction is shown in green, and then the wind speed is still shown in blue, because the wind speed and direction sensor remains in the same location, but the storm moves. So moving forward, I will be looking more at infrasound. So infrasound is just sound that is below the human hearing range. So many things can produce infrasound, including major weather events and earthquakes, tsunamis. So I actually do have infrasound recordings from these same 9 smartphones in Guam during the passage of Mawar. So ideally, the goal will be able to match these infrasound frequencies with specific frequencies that will be produced from different wave heights. So the idea behind this is that different wave heights have different frequencies associated with them. And if I can look at the infrasound recordings and extract the most dominant frequencies, I can use that and correlate them back to the wave heights. So that if that is successful, which might be optimistic, this can have other applications in the future for other wave events, maybe tsunamis or other things like that. And that is all I have. And I also just want to take a minute to thank my mentors, Eric and Ray, and everyone else at ESSD. It's been great working with all of you. So thank you.
Right. Do we have any questions for Hannah?
For those of you that don't know, Hannah's an oceanography student, but we had her looking at meteorological data. How was that transition for you during the summer?
It was good. I think the atmosphere and ocean have some similarities, so that's pretty nice, but it's not like night and day different.
So I quickly looked at the infrasound recorder app, and is it just something that if you have on your phone, it collects data and it aggregates it? Is your thinking maybe to encourage people in tsunami or hurricane prone areas to download that as background information so you can more accurately predict? Is that?
Yeah, I mean, I think that's the eventual goal, but so far it's in the earlier stages. So these 9 that I looked at were like very specific phones that were left in certain locations and were not touched. But I think that hopefully one day the goal would be to just have people have it on their phones to be of use.
Nice job on the presentation. I was wondering with the phones, is there a public database in which we could like access the information on those phones?
I think there actually is. Okay. Not completely sure, but you can. It's just like redvox.com.
Oh, okay.
I think is the website, and there's a bunch of information on there.
Do they have historical data as well, or is it just present? Like, is there any archival?
I'm not sure.
Just kidding. Thank you.
Sorry.
You have a question?
Just, following that idea, is it possible that at some point you could have an app on a phone that could basically collect and respond to that data and give you some sort of an alert that, something's coming or?
I hope so. Yeah, if this progresses in the correct direction, that would be really cool to see if we can make that happen. Yeah.
Any other questions? No? All right. Great job, Hannah. If we have enough time, Allie, do we have time for Kimberly to come show us the dashboard?
Yeah.
Can we do
Yeah,
that?
we're a little ahead of schedule. Oh, okay. I thought someone was speaking. Wait, real quick. We have a mentor speaker.
Oh, sorry. Hold on.
But I think did Haley step outside? Haley would be the best person you want to drive? She's good with the app.
Go ahead. We'll get you in. Yeah.
Right on. So thank you. Thank you, Hannah. I just want to say that we're all very, very proud of you. We got the data about 2 weeks before Hannah got here. And we thought, well, why not just throw this at her and see what she does with it? So she's been really, really good at it. As you can see, she's a true budding scientist. Over the past few weeks, she's really learned how to program in Python. She's honed her skills into that. And then as we start looking more and more into the infrasound information, she took the initiative to learn harmonic analysis and fast Fourier transforms. So she's really advancing in her advanced calculus knowledge as well. So really, really proud of you, Hannah, and thank you for all the work that you've done so far.
Or is that going to turn off? It will turn. It's much more fun in person, in live too. You can see the remnants of the storm that just passed. Did Calvin die? It's over here. If you went over there, you'll see. I'll let her drive. She's with better. Yeah. Oh, this is Maui. Is in the Simran? Yeah. In the back. I don't know how to do it. Oh, yeah. Okay, so you got it in. Why not just share the screen? Oh, but instead of that, I'm already logged into the meeting. I could just share the screen. Oh, but that was a okay. How do I do that? Oh, yikes. We're in the meet. And then do it better. Full screen, yeah. Do the whole screen. Oh. Share the tab. Pick the dashboard. This one. Okay. Oh. Hey. Yay. Grab us through and just this microphone should pick you up. Oh, okay. So hello again. So right now I'm going to zoom in on this cyclone so we can see more of its data. But right now what I was trying to show you guys through the presentation was that there was a sidebar over here that you can click to see how to use the dashboard. And then these were the links you could use to learn more about the different stuff at NOAA. And then right now here's a main cyclone by the Atlantic. It looks like Calvin died on us. But anyways, this is a gauge where it shows us the different strengths of the cyclone over time. So as we see, Don is getting slightly stronger. And yeah. And this is his most current. He's at 45 knots. This little indicator right here tells us how many active cyclones are in the area. So right now we're in the Atlantic, so we only see one. But if we zoomed out and the Calvin was still alive, you would see two. This one in the middle is just research vessel data about the certain ships. So we chose to keep speed, latitude, longitude, barometric pressure, and wind speed. Over here are the different ships and what they actually look like. And over here on all the top
right corners, there's a hover where you can expand on all of them. So we can also expand our map. Right here, we can zoom out. Right now we have our weather data layout layers open, which shows us the radar and weather pattern, but we can also turn it off with the eye icon, and it will just disappear. Same with the cyclone. However, the weather data also shows the cyclone. Yeah. But we can zoom in over here to Hawaii and look at the buoys layer. These are all the different types of buoys. Here's the layers names. One of the main important things we wanted to show were the ship's layers. However, that's still in a working progress to make it more easier to find. All right, now we can zoom out and look at the ship track layer. So right now, that pink line is SETI and that yellow lines are the Rainier. We can turn on its current location and it says the Rainier's over here. And then the SETI's supposed to be over here, but it stopped logging its actual data. Yeah. And there's some more of the different vessels here. What's this? This is Fairweather. Bel-Em Shimada. It's over here. And then Fernander. But yeah. All these little widgets over here is for like searching stuff. Which can take you to the exact location. There's bookmarks to scroll through different islands. So we can go to Guam. Takes us to Guam. Takes us to any of these bookmarks here. It can take us to America Samoa, which is where we can see the Rainier right now. Then we have a pull-up legend bar right here, which shows the different current ship locations and what all the cyclone symbols mean. And this is all the layers you can pull off and turn on. And you can also change the base map if you're interested in doing that. You press this and it will change the background. Yeah, that's pretty much it.
That is awesome. Thank you, Steph. That's very cool. I'm glad that. Yeah. And that's accessible now. There's a QR code for it.
Everyone with the OJAC will be able to see it. OMAO gives us permission. But if anybody in the group wants to try it out, just send us an email.
Great. Okay, and for those of you folks online, Kimberly said if you want to send her an email, if you want to test it out, to go ahead and do that to be added to the list. And just out of curiosity, are these presentations available anywhere after today?
There will be recordings. So the recordings will be going out to everybody so that you guys, if you have interest in looking back at yourself and seeing how your talk went and reliving it, you can do that. Or if you missed some of the other talks for any reason, those will be available and we can also share them with staff. So for those of you that don't know me, I'm Allie, and I just wanted to say a few words to close things out. First and foremost, thank you so much to all of the interns for all of the hard work you guys have put in all summer and then for your willingness to share that with us today. It's always very inspiring for us that have been around for a while to like get the, the new, fresh, young energy and just to learn about what you guys have been up to. It's always really inspiring. So thank you. I would also really like to thank all of those volunteers from across the IRC that helped make today possible. Not everybody is here right now, but just so you guys know, we've had Alex and Jim in the back pretty much all day, holding down the tech side of things. That is not an easy job, and we definitely had a few little hiccups that we had to deal with. And also we had, Andrea setting up the snacks, and Beth helping out, and Kaylin doing all of your moderating, and all the moderators, timekeepers. Thank you guys so much for helping out today. And I would like to invite Phoebe up just to say a quick thank you. So Phoebe has been instrumental in helping today work. She planned all of the practice runs and helped with all of the Google Meet stuff. So I just wanted to give you a huge thank you. And we're gonna invite. Oh, thank you. Wait, that's bad luck. It's bad luck. Sorry. Really? It has to be open. It's okay. I'm not superstitious. Thank you. And with that, I'll invite Ray up for a few closing remarks. Yeah, sorry. I didn't mean to be like, I took it off. It's bad luck to wear clothes lay when you're pregnant.
All right, aloha everyone. My name is Ray Tanabe. I'm the director for the National Weather Service Pacific Region. First and foremost, I just wanted to take the time to thank all the interns here in the room for your interest in working with NOAA this summer. I also want to thank all of the mentors who provided a lot of time and energy in working with you. Turned out some great projects. Many of you have traveled really far from home or school to be here with us this summer. And at the very least, I hope outside of your internship hours, you got to get out and enjoy some of the wonderful things that the islands have to offer. Whether studying sand abatement in Kihei, studying seal behavior, looking at different weather events, longline fishing, corals, all of the work that you did this summer contributes towards NOAA's mission of science, service, stewardship. One common theme I saw throughout the presentation today was the importance of data and observations in weather and just about in any science, right? Everything starts with robust observations and robust data. When I was in college, one of the things that my professors paid me to do was we looked at historical rainfall records in Hawaii, going back over 100 years, but they were all handwritten in journals kept by ranchers, kept by sugar plantations. And we spent hours in the lab writing those down, quality controlling them, and then putting those into an Excel spreadsheet so they could be analyzed further. I heard some stories about handing over dusty copies of records that go way back when and having to incorporate all of that. Don't ever forget the importance of good observations. And after all, once you've graduated and you've moved on and you're conducting your own research, just think that there'll always be more summer interns to do the dirty work for you. I'm amazed with all of the work that you have presented today, and I very much hope that all of you join the NOAA family down the road. One of my favorite slides was from the presentation on coral reef curriculum that was presented. And in that slide, there was a picture of Laura teaching these elementary school students about corals using edible coral polyps, right? It made me hungry, but that's awesome, right? So in my mind, all the connection was there. You had Patty Miller mentoring Laura, who was also teaching arguably the next generation of NOAA scientists. So it's all connected, right? It's all there. So that type of connection between all of the generations will ensure that NOAA's legacy of environmental stewardship continues well into the future. And that's inspiring. Can I get a show of hands real quick? Who here was born after 1996? After 1996.
Wow.
All right, it's a little embarrassing. But in 1996, I was in your shoes. It was my first summer internship with NOAA. I worked for the Office of the Federal Coordinator for Meteorology in Silver Spring, Maryland. And this is way back when, right? 1996. The internet was still relatively brand new. And what I did that summer as my project is I created the first ever website for this particular office. And I digitized a lot of material that they had that was only available in printed form. And so it was a lot of scanning of materials, a lot of digitizing of pictures, but that's what I did way back when. I continued to be a student intern with the National Weather Service Forecast Office here at the University of Hawaii until I graduated with my master's degree in 2000. From there, I steadily progressed through the ranks and I became the director for the Pacific region in 2013. And while I never imagined that I'd be in this position when I started in the National Weather Service, it's provided me with such amazing opportunities that I never thought I would have. And I'm forever grateful for that. Please keep in touch with us. Please keep in touch with each other. We can't take care of this earth by ourselves. We have to do it together. And finally, in 6 to 8 years, I plan to be retired. My goal is to be like one of the Honu in Jessica's presentation, right? I'm going to be basking on a beach somewhere, drinking beer and going fishing. But after today's presentations, I'll be able to do this confidently, knowing that NOAA's mission of science, service, and stewardship is well taken care of by folks like you here. Thank you.
All right, thank you guys so much, and feel free to grab some more snacks on your way out. And if I can get all the interns out into the atrium, we're gonna get a group photo. Thank you guys. Oh, thank you. And I also